So hey students, welcome to Vedantu Neet English. This is Basavaraj sir, your biology master teacher. Students, before we start, quickly let me know in the chat if my voice is audible to you and the presentation behind me is visible to each and every one of you. So quickly, good morning in the chat because we are starting early today. And students, this is one of those chapters which is actually difficult for some students. That is, quickly everyone raise your hands if this chapter is difficult for you. Quickly raise your hands if you do not understand the concept of, sir, what is this restriction endonucleases? Sir, what is this cloning vector? What is PCR? What are the steps in your biotechnology? What is an ideal vector? So if you do not know any of the following terms, don't worry. Do not worry at all, because in today's session, students, in today's session, we'll be covering everything. We'll be covering every single concept from scratch. And I will make sure you understand this chapter, and after today's class, you will not even have a single doubt in this chapter, and you will be able to solve every single question from this chapter. So are you ready for this beautiful class of biotechnology? So I'll be taking principles, then Gopika mam will come and she will be doing the application parts. Okay, ready students? So students, I'll tell you one thing before we start. Stay till the end of this video, because it will actually benefit you and you will not have any doubt regarding this chapter. So quickly smash that like button right now. Quickly smash that like button and we will start the chapter of biotechnology. And remember, today we will have killer visualization. When I say killer visualization, I mean proper diagram and live videos of your gel electrophoresis, live videos of so many experiments. So are you ready to learn biotechnology like never before? So are you ready? Yes, everyone in the chat? Quickly, I want some extreme high energy today. Extreme high energy, because we have a lot of stories to learn today. Okay? So this is our Telegram channel, that is Vedantu Neet English. You can scan this and you can join the Telegram channel. Now students, can anyone tell me what is the meaning of biotechnology? Anyone? So what is this bio and what is this technology? Very simple. Very, very simple. Bio means living. Yes, sir. Bio means living, and technology means some kind of technique. Some kind of technique. So we are using living organisms and we are applying some technique on it. We are applying some technique on it, like engineering type. Okay. Now when I talk about living organisms, I talk about cell, I talk about tissue, I talk about a particular life process, and I talk about your DNA and RNA. Every single one is a living tissue, and we are using some technique on this living tissue. Okay, we are using some technique on this living tissue. Now students, the question arises, why? You might be asking, sir, why do we need to do biotechnology? What is so importance of this biotechnology? What is so importance about it? Now let me tell you students, not just biotechnology, any technology in this world, any technology in this world is for human benefit. You'll agree with me, every single class is for every single particular technology is for human benefit. Can I give you a quick example? Yes. So how do you call your friends? Can anyone tell me in the chat? How do you call your friend? You might take your phone and you're like, how are you doing, bro? You call. Or you text your friend, right? Or you come on YouTube, or you text in the YouTube chat. You think I've not noticed? You text in the YouTube chat. But back in the days, how do you used to do? They used to write letters in the back in the day, right? I'm not talking about love letter. I'm talking about normal letter, not love letter. Normal letter they used to write and they used to send. If you go even back, they used to send a pigeon, like kabutar jo, right? Kabutar ja, they send pigeons. Even back in the day, in Akbar Birbal time, they used to send people for, transferring messages. But as the technology advanced, we have upgraded ourselves. So similarly students, biotechnology also, yes, similarly biotechnology also is for our own benefit, right? It is all for our own benefit. Students, am I audible? Yes, can you tell me in the chat, am I audible once again? Or should I increase my mic sound? I do not know how to increase my mic sound, but am I audible? One second. Check. Am I not audible? Sound check now. Audible now? or not audible still? Tell me in the chat quickly. Am I audible? Little bit more. Little bit more. Sound increase. Oof, oof, oof. Okay. I will try, but I do not know if I will succeed. Okay, okay. How do I increase? I think this is the maximum sound. Is it fine now, students? Check once again. Is it fine? Clearly audible to all of you? Yes. Okay. Now, where was I? Now, I was talking about biotechnology. Now, you might be thinking, audible, still audible, or do I need to increase more? We can hear, you can hear me, right? Use some technology and hear me more. Can you hear me? Increase your volume, right? No, not audible. Still not audible. Oh my God. 1 second, students. Let me call my friend. I'll use technology and I'll call my friend. Audible, right? Yes, I'm audible. Okay, students. Now, you might be wondering that is biotechnology a very new concept or is it a very old concept? And you might be wondering, sir, it sounds so fancy. It must be born yesterday only. You must be born yesterday only. No. Students, remember, biotechnology is actually very old. It is very, very, very old concept. For example, even your mother is using biotechnology. Even your grandmother is using biotechnology. Even your great-grandmother is using biotechnology. Now, you might be asking, how, sir? How is my grandma using biotechnology? Right? You might be asking, how is my grandmother using biotechnology? Can I tell you a simple answer? Yes. Have you noticed your mother making curd in the house? She will take a small curd and she will add it in the milk, and the next day the milk will become curd. That is, your mother is taking a small amount of bacteria. Can anyone tell me which is that bacteria? That is your lactobacillus. All of you know it as lactobacillus. So your mother is taking a little bit of bacteria. She is adding it into a medium. Now, which is the medium there, students? The medium is nothing but the milk. At right temperature, the lactobacillus is utilizing the milk, the media, and it is getting converted and it is multiplying. Yes. Now, how many of you like dosa? Can anyone tell me in the chat? How many of you like dosa? Because I love dosa. Right? I love bende dosa, and this is one of my favorite breakfast. My favorite breakfast. But have you noticed when your mother is making dosa, one day before, in the night time, when you are watching Big Boss, when you're watching IPL, suddenly from the kitchen, one gwee sound comes. Have you noticed? When you're watching TV, suddenly one sound comes. Gwee. You might be like, you'll start screaming, Mom, why are you making that sound in the kitchen? What is your mom doing there? Your mother is actually preparing one day before, in the night time, for dosa preparation. You might be asking why. Because students, remember, the dosa preparation also requires some amount of bacteria which will grow overnight and will become swollen. Right? Right, students. Now, one more example is there, students, which is all of your favorite. Which is the example? That example is your wine preparation. Wine preparation or cheese preparation. Wine preparation and cheese preparation is also a traditional method. Students, your wine preparation, curd preparation and bread preparation, all of this come under traditional biotechnology. It comes under traditional biotechnology, right? That is science of using living organisms and their products for benefit of humans and surroundings. Now, can I give you one more example of biotechnology, like traditional biotechnology? That is your selective breeding. That is your selective breeding. All of you know selective breeding, right? That is, we have a skinny chicken which lays more egg. Imagine it is going for an interview at KFC. Do you think this will get selected at KFC? No. You'll get rejected at KFC telling that it is very skinny. Rejected. Imagine we have one more chicken, but it is very hefty chicken, very fat chicken, but lays very few eggs. But will it get selected at KFC? Yes, it will get selected at KFC. Okay. Now, what do? You take the genes from you do a You do a hybridization. When you do a hybridization, whatever desirable gene was present here and whatever desirable gene was present here, by chance, it might come into a offspring. Now you will have a hefty chicken which lays more egg. But there is one problem. But there is one problem. What is it? Along with desirable genes, can the undesirable genes also come into the picture? Yes. Imagine if this particular chicken, even though it might look hefty, but the lifespan is very less. Right? So that is an undesirable gene which is coming here. Undesirable gene is also coming here. So that is the problem. That is the problem with your selective breeding. Yes. Now students, do you remember 2 years back what happened on Earth? Yes, 2 years back what happened on Earth? It was a small disaster, right? Serious mode. 2 years back there was a disaster on Earth. That is a COVID came into the picture. Some of you might be happy in the house, like dancing, oh, PUBG whole day, 10th are done, no class 10th examination, 10th class done and dusted without even studying. You might be thinking that time, in the COVID time, all of you are enjoying in the house. But do you know, there were some heroes wearing a white coat, working day in and day out. I remember my mother, who's a nurse, who did not come to the house for at least 1 month. She did not come to 1 month to the house. I remember, because it is so difficult for doctors and nurses that time, right? Which you are also trying to become a hero that day. You are also trying to become a hero, right? In that condition, how did we overcome? How did we overcome that situation? Does anyone remember? Does anyone remember how we overcame that situation? It was by vaccination. It was by vaccination. And do you know, this vaccination is made with the help of biotechnology. It is made with the help of biotechnology. So can everyone in the chat type biotechnology OP? Can everyone in the chat type biotechnology OP in the chat? Because without biotechnology, we would be still in lockdown. We would still be in the houses. There would be so many deaths outside. So that is the importance of biotechnology. And this chapter, students, this chapter, because this is one of those chapters which has so many practical applications which you need to study, not just for marks. But do you know how many marks this chapter has? Approximately 44 marks, including the application. 44 marks. So can you afford to miss this chapter? No, you cannot afford to miss this chapter. And this is one of the chapters which will teach you life also. So can we start this chapter, students? Students, this vaccine, which first vaccine was given by Edward Jenner. Then we have antibiotics which all of you take, right? Antibiotics which all of you consume. We have the penicillium, right? All of this, along with your IVF techniques, in vitro fertilization, test tube baby, everything comes under your modern biotechnology. All of this comes under modern biotechnology. Clear, students? That is, traditional biotechnology, both desirable and undesirable characters, but in modern biotechnology, only desirable traits are used. Okay? Now, can we start the class with a simple definition? The definition given by, not by me, not by you, the definition given by your European Federation of Biotechnology. Now, what is the definition? The integration of natural science and organisms, cells, parts, therefore, and molecular analogs for products and services. That is, we are taking advantage of your natural sciences, traditional biotechnology, modern biotechnology, and for products and services. Now, what is this molecular analog? Molecular analog is nothing but your DNA and RNA. DNA as well as RNA. Okay? Now, can we start the chapter with principles of modern biotechnology? We'll start the chapter with principles of your modern biotechnology. Now, what are the principles, students? The entire biotechnology can be divided into two categories. The entire biotechnology can be divided into two separate categories. The first category is called as genetic engineering. Now, what is happening in genetic engineering? Listen very carefully, all of you. Now, imagine this is a segment of DNA. This is a segment of DNA. Now, from this segment of DNA, students, from this segment of DNA, imagine I'm trying to cut a piece out. This is a particular gene responsible for a particular character. Now, how am I cutting this? I'm able to cut this particular segment of DNA with the help of a magic scissors. With the help of a magic scissors, which we'll talk about it later. Okay. Now we have a magical scissor and we're cutting it. Now we are removing this particular DNA. We are adding it into a vector and we are able to replicate this. We are able to replicate in a lab. Now quickly tell me in the chat, is this in vitro or is it in vivo? Is it in vitro or is it in vivo? So all of you tell me, sir, it is in vitro in nature because it is happening in the lab. It is happening inside the lab. Now students, if it is happening in the lab, do we need a proper sterile environment? Do we need that? Yes. We need a proper sterile environment. Yes. Sterile environment. To maintain this sterile environment, to make sure only and only our desirable microbe is growing and nothing else is growing. That is called as your bioprocess engineering. That is called as your bioprocess engineering. The cutting of DNA, making the recombination, all that comes under genetic engineering. But maintaining the sterile condition, making multiple copies of your gene of interest, making multiple copies of your organism, all that comes under bioprocess engineering. Bioprocess engineering. Okay. See here, what is genetic engineering? Techniques to alter the genetic material of an organism to change its genotype and phenotype. Genetic engineering, that is, we take a gene, we cut this gene is responsible for protein, we add a modification to this gene and we get a different protein altogether. That is, we are modifying a gene to make sure we get a different phenotype. Clear? That is your genetic engineering. Genetic engineering. Now what is bioprocess engineering? Maintenance of sterile conditions to enable the growth of only desirable microbes to obtain pure products. That is your bioprocess engineering. Does anyone has any doubts? Quickly tell me in the chat. Does anyone has any doubt difference between genetic engineering and bioprocess? Tell me in the chat. See here, bioprocess engineering is multiplication of your GMO as well as your product formation. If no one has any doubt, quickly solve this question, all of you. Quickly solve this question. And also quickly smash that like button because this is how the chapter is going to be. This is a very interesting chapter and I want it to be memorable for all of you. Okay, solve the question. Sterile environment has to be maintained in bioprocess engineering. The term sterile in microbiology and bioprocess engineering refers to inability to reproduce. What is it? Tell me in chat quickly. All of you know this is assertion is true, but reason is false because we know sterile condition means a particular condition in which no other organism is able to grow. No other organism is able to grow. Clear? Now, students, can I give you an overview of this chapter? Can I give you a complete overview of this chapter with the help of a small story? Yes, small overview. What you want to study in this chapter? What are the different processes which we have? Can I start with a small character called as Pandu? All of you know Pandu, right? The famous character Pandu. Let us draw Pandu here. Yeah, this is our Pandu here now. We have Pandu. Okay, we have our Pandu now. Now Pandu has a best friend. Can anyone tell me the best friend of Pandu? Pandu has a best friend, but his best friend is not Champa now. He has one more male best friend. His name is Macha. We have a new best friend now in the house. Nice hairstyle called as Macha. Okay. So we have Pandu and we have Macha here. Now imagine, students, Pandu is around 6 feet tall. Pandu is 6 feet tall. Okay. Pandu is 6 feet tall. But Macha is around 4 feet tall. Macha is just 4 feet tall now. Macha is 4 feet tall. Your Pandu is 6 feet tall. Very tall person, Pandu. Okay. Now students, listen to me very carefully. Now, Macha is like, Pandu, how did you become so tall? How did you become so tall? I'll write the name also, all of you will forget. Now Macha is asking Pandu, how are you so tall in life? Now Pandu will tell, no, I am not doing anything. It is in my genes. It is in my genes that I am so tall. So now what will Macha do? Macha is very curious. Macha is very jealous. Now what will Macha do? When the Pandu is sleeping, when the Pandu is sleeping nicely, Macha will go to his house. Macha will go to his house. From his hand, he will take out one small cell. He'll take out a small cell. Okay. Now imagine this is the cell he took out. This is the cell he took out, and inside the cell, students, inside the cell, he got the DNA. Inside the cell, let's make it a small so we can write entire story. He found the DNA. He took the Pandu's DNA. Now students, inside the Pandu's DNA, do we have the gene responsible for tallness? Yes. We have a particular gene for tallness. Tallness gene is there. Now what did you do? Now this particular gene is there, right, inside this DNA. Macha, what did he do? He took a scissor. And he cut it. He took a scissor called as molecular scissor. He took a scissor called as your molecular scissor, which is called as your restriction endonuclease. It's called as what? Restriction endonucleases. Now what is this restriction endonuclease, which is also called as a molecular scissor? That I'll tell you later on. Don't worry for now. Now students, when the Macha took a scissor and he cut both the ends, he got a particular segment of DNA. He got a particular segment of DNA. And we call this particular segment of DNA something called as your desired DNA. What do we call it? Desired DNA. Can I write here? Desired DNA. Desired DNA. What is the other name for this desired DNA, students? The other name for this desired DNA is called as alien DNA. We call it alien DNA. Now students, we take this alien DNA and we add it into a something called as a taxi. We add it into a taxi. Now this taxi is nothing but something called as vector. What do we have now? We have the vector here, students. We have the vector here. We have the vector. I'll give you a quick example, students. Quick example is going to be your plasmid. Quick example is going to be a plasmid. Now what do we need to do? What do we need to do, students? Okay, if you want to make this alien DNA into the plasmid, we need to cut the vector also. Yes, we need to cut the vector. Okay, fine. We'll cut the vector here. Not we. Who is doing all this work? You are doing. No. Macha is doing all this work. Okay, Macha is doing this. Macha is scientist. You don't know. He cut the DNA here. He cut the vector here. Vector DNA. Now what did he do? After cutting the vector DNA, he added the desired or alien DNA inside. He added the alien DNA inside. Now this vector DNA has a new name. This vector has a new name. What is the new name, students? Can anyone tell me in the chat what is the new name? The new name is something called as your recombinant DNA. It becomes what? Recombinant DNA. It becomes what? Recombinant DNA now. Students, now this recombinant DNA has to be transferred to Macha. Yes, because that's what he's doing all this work for. Because he wants to grow tall. That's why he's doing all this work, right? So what we do now is, we take this recombinant DNA and we add it into our host. Who is the host here, students? Macha is equal to host. Add it into the host now. Now, this process of adding recombinant DNA into the host is called as, your transformation. Transformation. It's called as what? Transformation. And once we add the recombinant DNA into the host, this becomes transformed. It becomes transformed. So now we have what? Now we have our vector in recombinant DNA. We have a recombinant DNA inside the host cell. Now do you think, students, Macha will become tall? Yes. Macha is like, finally I'm so tall. Macha is like, finally Macha will grow in height. Macha will also become 6 feet. Students. Now, if Macha is becoming 6 feet, do you think Pandu will become short? Do you think Pandu will become 4 feet? No. Just because Macha became 6 feet, Pandu will become 4 feet? No. Because students, remember, this particular gene which is in the DNA, it is present in every cell. It is present in every cell. You cannot be like, just because he became tall. No. No, no, no. So both are tall now. Happy ending. Done. So what do you understand this story, students? What do you understand this story? In your biotechnology, students, in your entire biotechnology, what is the first step? Can anyone tell me which is the first step? The first step is going to be identification of gene of interest. Identification of gene of interest. So that is your first step. Here, where is the first step, students? Here is the first step. Did we identify our gene of interest? Yes, we identified our gene of interest. The second step is making your recombinant DNA. Second step is your making the recombinant DNA. Yes, done. Recombinant DNA we have. The third step is going to be your transformation. Third step is going to be transformation. Done. Transformation also done. And also, once transformation is done, this particular vector, will it multiply inside? Yes, it will multiply inside. That is technically your fourth step. But in general, we consider only 3 steps. In general, we consider only 3 steps. Are you understanding, students? Are you understanding? With the help of a small story of your Macha as well as your Pandu, you have decoded the entire chapter. Good night, bye-bye. Just kidding. Just kidding. So students, this is your entire chapter. This is your entire chapter. Now students, can we go one by one for every single step? Can we go one by one to every single step now? Okay. Now students, let's understand the recombinant DNA technology now. Let's understand recombinant DNA technology. Now what is this? Technology used for introducing DNA sequence from one organism to another organism. Here, this is your DNA technology. From one organism, you're introducing the DNA into another organism. That is your recombinant DNA technology. That is your recombinant DNA technology. And do you know who is the scientist? Do you know which scientist made this? That is your Stanley and Cohen as well as Herbert Boyer. As well as Herbert Boyer. These are the 2 scientists who made the first recombinant DNA. And can you tell me which year? This was done in your 1972. Was I born in 1972? No. Were you born in 1972? No. But do we have to study this? Yes, we have to study this. Okay. Now, what did they do? What did they do? Are you ready for one more story now? Are you ready for one more story? Yes, sir, we are ready for one more story. Now, students, unlike Pandu and Macha, they worked on a bacteria. They worked on a bacteria. That is your Salmonella typhimurium. Salmonella typhimurium. Okay, Salmonella typhimurium. Now they noticed, oh my God, in the Salmonella typhimurium, there is a special antibiotic resistant genes. They have what? They have antibiotic resistant genes. They're like, wait a minute, can we take advantage of that? Yes, we can take advantage of that. So what did the students? They took your Salmonella typhimurium plasmid. Oh, it has gone to the side. Okay, wait, let me draw it for you. Doesn't matter. We can draw. Yes, we can draw. This is your plasmid. This is your plasmid with your antibiotic resistant gene. We have what do we have here? We have the antibiotic resistance gene. We have the antibiotic resistant gene. Now, remember in the starting, students, remember in the starting I told you, I can cut it. Remember I told you, molecular scissors. We can cut this antibiotic resistance gene with a molecular scissor. Now, can anyone tell me in the chat, what is this molecular scissor and what is its origin story? Can we go to the small origin story of this molecular scissor? Yes, we can. Students, remember this molecular scissor. This molecular scissor is called as your RE. That is nothing but your restriction endonucleases. Restriction endonucleases. Now, how do we obtain this restriction endonuclease, students? this is obtained from your bacteria. This is obtained from your bacteria. Okay, that is mainly your prokaryotes. This is your bacterial cell. Now, imagine this is your bacterial cell. This is its genome. This is the nucleoid, right? This is the nucleoid. Now, imagine one bacteriophage is entering. One bacteriophage is taking entry here. Okay, one bacteriophage is taking entry. It is releasing its genetic material. Yes, it is releasing its genetic material. Now, students, imagine if a thief come to your house, will you be quiet? You're like, no, take my money and go. Will you tell the thief, take my money and go, take my gold chain, take my iPhone, take my gold watch? There is in the kitchen, in my kitchen, my mother in the one small box has kept money, take that also and go. Will you tell that? No. You will fight against the thief. Yes. So similarly, your bacterial cell will also fight against the genetic material. It will fight against the genetic material. How? This particular bacteria produces restriction enzymes, and this restriction enzymes completely chop the genetic material. Completely chop the genetic material. Done. Can this bacteriophage multiply inside? Can it multiply inside? No. Not also multiply inside. Now, students, imagine, can this restriction enzyme attack its own genetic material? Can it attack its own genetic material? No. Now, why? Why is the guarantee that it won't attack its own genetic material? The answer is very simple. That is, this genetic material, this particular genetic material is very smart because it has something called as your methyl additions. Methyl additions. Now, if this methyl additions is there, because this methyl additions, restriction enzyme cannot attack its own genetic material. Okay, cannot attack its own genetic material. Done. Okay, now can we go back to the story? Can we go back to this story now? Okay, going back to this story, listen to me very carefully. The two scientists, okay, they took the plasmid of your salmonella. They cut the restriction enzyme. They cut the antibiotic resistant gene. They cut the antibiotic resistant gene. See, restriction enzyme is coming and it is cutting the chopping the antibiotic gene. Okay. Now we took one more plasmid. Now what did we do? Here also we took a piece. Now we took the vector plasmid. To this vector plasmid, we added our antibiotic resistant gene. And then we pasted the two different DNA segments with the help of a glue. The glue is called as DNA ligase. Now, once we added the glue, two different DNA fragments, they joined. Now, we took this recombinant DNA. Remember I told you starting, we take a vector. To the vector, we add a desired gene and it becomes your recombinant DNA. Now, we take this recombinant DNA, we add it to the host. Yes, we take this recombinant DNA and we'll add it into the host. Now, who is the host here? Can anyone tell me? Who is the host here? The host is going to be your E. coli. So, once we add our, can you see the difference color here? Once we add our recombinant DNA into the host, this is called as your recombinant E. coli. Not normal E. coli. It is called as what? Recombinant E. coli. Can I quickly summarize the entire story? First, we had the plasmid with antibody resistance. We chopped with the help of restriction enzyme, we cut the antibiotic region. Now we have the antibiotic resistant gene is here. Now we took one more plasmid from same salmonella only. We took one more plasmid, that is your salmonella plasmid, which is acting as vector. Now we have your fragment of salmonella plasmid and we have the antibiotic resistant gene. We added the dose two. Yes, when we add your vector plus your antibiotic resistant gene with the help of gum, that is your DNA ligase, we get our recombinant DNA. Yes, we take this recombinant DNA and we add it into the E. coli. And as the E. coli is multiplying, we obtain our multiple copies. Clear? Clear, students? Clear? Are you understanding? That is your experiment done by your two different scientists. Where are the? Let me show the images because NCR doesn't show you image. This guy looks more handsome, no? Huh. So Stanley N. Cohen as well as Herbert Boyer. They were the two scientists who did the experiment. Clear? Concept is getting clear? Concept is getting clear, right? That is more important. That is more important. Okay? Concept should be important. How this thing is happening. If you just read the NCERT, it is a little difficult to understand. Little difficult to understand. But when you have killer visualization, like this, does it become easy to understand? Yes, it becomes easy to understand. That is why this chapter is more important. Okay? Now students, now can we go to the steps of rDNA technology? What are the different steps in your rDNA technology? Can we? Gopika mam is in the chat. Yes, I didn't see that. Sorry, ma'am. Okay. Now, what are the steps in your rDNA technology? Let me tell you. I'm here. Don't worry. First one is your identification of DNA with desirable genes. Yes. In your Pandu and well as your Macha, did we identify your gene of interest? Yes, we identified a gene of interest. Introduction of identified DNA to the host. Did we take that DNA segment and we introduce in the host? That is called as transformation. We did that also. Maintenance of introduced DNA in the host and transfer of DNA to its progeny. So Macha's daughter and son will also be tall now. Macha's daughter and son also will be tall, 6 feet. Now students, can we solve one simple question now? Simple question? Yes. Now what is the simple question, students, here? Solve it. Take your time. Let's see if you have marked your attendance. Oh, you have not marked your attendance. Students, do you realize how beautiful we are doing the chapter? How beautifully we are approaching a chapter. This is such an easy chapter if you understand with a visualization. This is a chapter where you will not even lose a single mark. You will not even lose a single mark. Okay. In recombination DNA technology, the term vector refers to a plasmid used to carry DNA into living cell. Yes. Yes. Done. Easy. Understanding? Understanding, that is your vector. Okay, that is also called as your taxi. Okay. Now students, let's go to the next concept. Tools. What are the different tools in your rDNA technology? There are so many tools. Let's understand each one by one. Until now, if you have any doubt, ask me in the chat. I'll clear it out also. Now first is the tools is going to be your restriction enzyme, which we just learned. That is your molecular scissor. There is DNA ligase, that is your gum, vector, taxi, and your host organism, that is your macha. Okay, these are the different tools which you need to use in your recombinant DNA technology. Different tools are there. Okay, now the question of the hour. The nation wants to know, how do you cut the DNA? How do you cut the DNA? Spoiler alert, all of you know, it is restriction endonucleases. But when I say students nucleases, when I use nucleases, you need to remember it is a type of, it will cut your nucleic acid. Nucleases break down nucleic acid. So based on that, we have classification of nucleases, students. We have two types of nucleases. One is called as your exonucleases. The other one is called as your endonucleases. Now students, what is this exonucleases, students? Listen to me very carefully. If this is your DNA fragment, and your molecular scissor is cutting on the terminal ends. It's cutting on two different terminal ends. That is called as your exonucleases. Now imagine, we do not want this. No. We do not want this. We do not want exo. We want endonucleases. Now what is happening in your endonuclease, students? In your restriction endonucleases, if this is your piece of DNA, your endonucleases will cut right in the middle or a little away from the middle. Right in the middle or little away from the middle. That is very important. Middle or away from the middle. Okay? Middle or away from the middle. See here, cut the DNA at the ends of which DNA molecule makes cut within the DNA molecule. DNA molecule. Now, if there is no doubt in that concept, can I teach you one more concept with your permission? All of you? Yes. All of you will be like, oh, when will you say no? Obviously, you will say, all of you will say yes. Now, students, your restriction endonucleases. Restriction endonucleases can be further divided into two parts. It can be further divided into two parts. Sir, we can divide into two parts. Now, what are these two parts, students? What are these two parts? Listen to me very carefully. Listen to me very carefully. If this is your DNA fragment, if this is your DNA fragment, I will call it DNA fragment. Now, here we have a sequence G, A, T, T, C, G, A, T, T, C, G, A, T, C, C. Yes. Now, on this DNA fragment, there is one restriction endonuclease. All of you know the name. Quickly spam that name. Yes. Quickly spam that name. What is the restriction endonucleases? We have a restriction endonucleases called as your ECO R1. We have a restriction endonucleases called as your ECO R1. Now, this ECO R1 is very special. This ECO R1 is very special. It will cut your DNA at a specific location. This specific location, students, this specific location is called as your recognition site. It's called as what? Recognition site. Yes, it is called as your recognition site. Called as a recognition site. Now, on this recognition site, there is a separate sequence called as your palindromic sequence. Yes, there is a separate sequence called as your palindromic sequence. Now, what is this palindromic sequence? Palindromic sequence is a sequence when you read from one side and the other side. Both are same to same. Yes. Both are same to same. Yes. Example I'll give you. Example is very simple. Example of a palindromic sequence is going to be your mom, dad, as well as the other word called as your Malayalam. Also called as your Malayalam. That is also your palindromic sequence. See, MOM, MOM. That is your palindromic sequence. See here, GAA-TTC, GAA-TTC. Is it a palindromic sequence? You will be like, yes, sir. It is a palindromic sequence. All of you know. Now, students, tell me in the chat. Will this restriction endonucleases, will it cut right in the middle? Will it cut right in the middle, or will it cut away from the middle? Tell me in the chat. Should I block the spammer? We have a spammer on the hand. No work. Early morning, no work. No productivity. Huh? So, students, it will not cut in the middle. It will cut away from the middle. It will cut exactly here. Can you see this? It will cut exactly here. Okay. Let me change the color, students. This much part is removed. This much part is removed. Now, students, listen to me very carefully. Listen to me very carefully. Now, what are we left with now? Can anyone tell me what are we left with? We are left with G here, C here, T here, T here, A, as well as A, as well as A. Now, students, I'll give you one more example. Can I give you one more example? Okay, 1 second. I'll give you one more example. Okay, listen to me very carefully. Listen to me very carefully. I'll give you an example now. Okay. When you are in a relationship, okay, when you are in a relationship, imagine your boyfriend or girlfriend will leave you and go. Will you become single again? Yes. Will you become single again? Yes. This is the single DNA now. This is a single DNA which is ready to mingle. Ready to mingle. Okay. It is ready to mingle. Single but ready to mingle. Okay. Now this particular DNA, this particular segment of DNA, is it ready to meet someone else? Yes. Is it ready to hang out with someone else? Yes. Can I tell this particular strands here, this particular different base pairs here, are they single here? Yes. Are they single here? Are they to hang out? Yes. Are they hanging here? Yes. So this particular segment of DNA is called as your hanging ends. They're called as what? Your hanging ends. Okay. They're called as hanging ends. They're also called as your sticky ends. They're also called as your sticky ends. They're also called as what? Sticky ends. Okay. They're also called as sticky ends. They're also called as your cohesive ends. They're also called as what? Cohesive ends. Okay. Since they are very single now, they're called as hanging ends. Okay. Or sticky ends, also called as your cohesive ends. Also called as cohesive ends. Okay. Did everyone understand? Did everyone understand? What is your restriction endonucleases, which cuts little away from the center. Okay. Now listen to me very carefully. Listen to me very carefully now. Okay. We have one more DNA sequence here. We have one more DNA sequence here. That is C, 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 G, G, G. C, 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 G, G, G. Okay. That is your DNA sequence now. Now imagine we have one more restriction endonucleases now. We have one more restriction endonucleases here now. Example is going to be a small one. Small one. Okay. Now this restriction endonuclease is very special. This restriction endonuclease is very special because instead of cutting away from the center, this restriction endonucleases will cut right in the middle. It will cut where? It will cut right in the middle. Right in the middle. So what do we obtain now, students? What do we obtain? All of you know. C, 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 G, 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 C, C, C. Yes. Do you obtain this? Yes, we obtain this. Now, but in this case, students, even though the breakup has happened, are they ready to mingle? No. They're not ready to mingle. They're like, no. Why? Because as soon as one guy approaches, the girl will be like, no. She will bluntly say, no, you're not my type. You might be thinking the worst she can say is no. But she'll be like, no. She'll bluntly give you answer, you're not my type. Okay. So, and that's why they're called as what? Blunt ends. They're called as what? Blunt ends. They're also called as your. Imagine she'll be like, nah. You're not my type. Go flush yourself. Okay. They are also called as flush ends. They're also called as flush ends. Okay. Also called as flush ends. Here we have hanging ends as well as sticky ends, also called as your cohesive ends. Okay. Blunt ends or flushy ends. These are two different types of restriction endonucleases. Did everyone understand? Yes. All of you are amazing. You will understand all the story, you will understand. When you say so many stories, you will understand. Without story, you will be like, nah, sir, give some more drama. Okay. Reminding of drama. You tell me in the chat, which one has more drama? You tell me, this has more drama or this one has more drama? You'll be like, okay, fine. This one has so much of drama because we have hanging ends here. So students, in your genetic engineering, we take more drama. And we take this type of hanging ends. We take this type of restriction endonucleases in our genetic engineering, in our biotechnology. Okay. So this is approved for genetic engineering because when we have hanging ends, students, it is very easy to attach your gene of interest. Okay. Or your vector DNA. Clear? Clear, students? Now, What is your restriction endonuclease, students? Restriction endonucleases is basically, restriction endonucleases are your enzymes found in your certain bacteria that cut the DNA molecule at or near the specific nucleotide sequence. That nucleotide sequence is called as what? That nucleotide sequence is called as your recognition sequence or palindromic sequence. Okay. Now, let's quickly summarize the discovery of restriction endonuclease, which I already told you. We have bacteriophage. This bacteriophage is entering here. Yes, bacteriophage is entering. Now, if this bacteriophage is entering, will this particular bacteria keep quiet? No, they'll not keep quiet, and they will come and they will release your restriction endonucleases. They will release what? Restriction endonucleases. That is, restriction modified system, restriction enzymes cuts the foreign DNA. All of you know this already now. So this restriction enzyme will come. Looks very cute, no, but it's very dangerous. Looks cute, but very dangerous. It will come and cut it. It will come and cut it. Now, what is stopping restriction enzyme from cutting its own bacterial DNA? That is modification enzyme. Modification enzyme will modify your bacterial DNA. So restriction enzyme cannot cut it. Restriction enzyme cannot cut it. Okay. That is your restriction enzyme as well as your modification enzyme, which are found inside your bacteria. Summary of the story. Okay. Now, restriction modification system was discovered in your E. coli. Restriction modification system was discovered in your E. coli. Clear? Now, action of restriction enzyme, all of you know already. That is, your restriction enzyme cuts the DNA at specific sites called as your recognition sites. Yes, recognition sites. What are recognition sites? A specific sequence which are recognized by the restriction enzyme to help and identify where to cut the DNA is called as your recognition sequence or recognition site. Okay, and remember, this point is very important. Very important. That is, more than 900 restriction enzymes have been isolated from over 230 strains of bacteria. 230 strains of bacteria. And what is the palindromic sequence? All of you know. Palindromic sequence which is read same from both the sites. Okay, all of you know this. And we can get blunt ends or we can get sticky ends. We can get blunt ends or sticky ends. Now, how do we get blunt ends? If DNA is cut in the middle, recognition sequence, we get blunt ends cutting. Blunt ends. How do we get sticky ends? Recognition sequence like your eco-R1 cutting in the away from the site, we get the sticky ends, also called as your overhanging ends or hanging ends. Okay, this is a different list list of different endonucleases which we can obtain sticky ends as well as blunt ends. Key blunt end, small one. Your sticky ends, we have HIND3, eco-R1, BAM-H1 and PST1. BAM-H1 as well as PST1. And remember, students, the first restriction enzyme discovered was your HIND2. It was HIND2. Okay, this is also given in your NCERT. Can you see? Till here, any doubt, ask me. Till here, any doubt, ask me right now. I'll clear. Did all of you understand what is blunt end, what is sticky end, the entire, how did we obtain restriction enzymes from your bacteria? All that concept is clear for all of you? Is all that concepts clear for you? If that concept is clear, can I teach you one more concept which is from your NCERT now? Can I? It is clear. All of you drink some water now. Yes. PDF will be provided, don't worry. PDF will be provided, don't worry. I'll give you the PDF, don't worry. Volume is still low, is it? Check. Oof. Is the volume still low, students? Is the volume still low? Clear, right? All of you are able to understand, right? Are you enjoying the session? Are you enjoying the session and making effort to understand this? How can I increase more volume than this? Let me check the volume on my phone. Volume is still low, is it? Check. No, volume is high only. I am speaking so loud there. Okay? Okay. Now, students, if you look at your NCERT, right now, action of restriction enzyme, that is, the enzyme cut both DNA strands at the same site. That is, we have a vector DNA we have a foreign DNA. Vector DNA and foreign DNA are cut by same enzyme. That is very important. It is cut by the same enzyme. Okay, please, ma'am, it's like use headphones. Vector DNA and foreign DNA both are cut by the same enzyme. That is EcoR1. All of you know EcoR1. EcoR1 cuts the DNA between bases G and A. Right? It cuts right here between your G and A. And what do we obtain? We obtain your sticky ends. Now this is your foreign DNA, that is also called as your gene of interest. Yes. And we have vector DNA. Now we take the gene of interest and we add it to our vector DNA. And what do you obtain? Fragments, DNA fragment join at sticky ends. And this joining is done by whom? The joining is done with the help of your DNA ligase. DNA ligase. Okay, DNA ligase. And finally we obtain our recombinant DNA. See here again, foreign DNA, plasmid DNA. We cut a piece here, here also we cut. Now what do? We take this cut piece and we join it here. And once we join, what do we get? We get a recombinant DNA molecule. Now we take this recombinant DNA molecule and we make it enter inside a cell. That process is called as what? That process is called as transformation. It's called as what? Transformation. Now later on this particular cell will divide and our gene of interest will multiply. Our gene of interest will keep on multiplying. Clear? Gene of interest will keep on multiplying. Students, this is a very easy chapter. I don't know, nothing is difficult. I'll make sure the entire chapter becomes very easy for all of you. Okay. Now students, let's quickly understand a little small concept called as nomenclature. Small concept called as your nomenclature. Okay. Now if you look at your entire structure, name of your bacteria, if you look at the name of your any type of your restriction enzyme, not bacteria, any type of your restriction enzyme, the entire name is divided into 4 parts. First, second, third, fourth. The first part is called as your part of your first letter of the genus name. The first part is going to be first letter of your genus name. Second part is going to be what? First letter of your species name. First letter of your species name. Third part is going to be strain type. Fourth part is going to be your order of discovery. Order of discovery in your Roman letters. Order in your Roman letters. See, example I'll give you. Look at EcoR1. Look at EcoR1. What is E? E is going to be your, from Escherichia coli, we are taking E. What is CO? CO stands for coli, that is species name. So CO from here. Now what is R? R is going to be your strain type, that is RY strain. From RY strain, the R is mentioned here. What about the 1? This is your 1, Roman letter 1. That is the order of discovery. That is your order of discovery. Clear? I'll give you one more example. That is your hint 3. See, H for your Haemophilus influenza H. From influenza, IN is there in N. D is going to be your strain type. And 2 is going to be your order of discovery. Order of discovery. Then we have DNA ligase tools we learned. DNA ligase is basically gum which will help us to fuse between 2 DNA fragments. 2 DNA fragments. Can everyone quickly solve this question now? Can everyone quickly solve this question? Which of the following is not applicable? Not applicable to plasmids. Not applicable to plasmids. Extra chromosomal DNA, always linear in form, single stranded molecule, autonomously replicating. Quickly in the chat. Quickly. Student, this chapter is very easy. Very, very easy chapter if you pay attention. Only if you pay attention, it's going to be easy for you. Unless you pay attention, it's going to become nightmare. It is a very easy chapter, students. Very simple chapter it is. Okay. Tell me. Not true. Not applicable. See, we know it is not single stranded and it is not linear. It is not single stranded or it is not linear. It is circular and it is double stranded, which is plasmid. Okay. Now, students, are you ready? Are you ready for the next concept that is called as your gel electrophoresis? Ready? Everyone in the chat? I want to see some green hearts in the chat if I want to know if you have understood every single concept up till now, and also if you are enjoying the session, and also if you are ready for your gel electrophoresis now. Ready? Quickly, green has in the chat, which will make sure I know that you are ready for gel electrophoresis. We'll take 2 minutes break and we'll talk now. Let's take a 2 minutes break and we'll talk. Now students, I'll ask you one question now. How many of you have done gel electrophoresis? How many of you have done gel electrophoresis? Tell me in the chat. How many of you have done it? Because I remember doing gel electrophoresis when I was in my class 11th. When I was in my class, younger than you. Yeah, younger than you. I went to IISc and I did gel electrophoresis for the first time. In class 11th, I did isolation of your prokaryotic DNA. Class 12th, I did isolation of your eukaryotic DNA. But if you have done the gel electrophoresis, you will know it is a beautiful process. But if you have not done this process, students, if you have not done this process, don't worry. Do not worry. Today I will make you do this. Today I will make you do this with the help of a video as well as with particular diagrams. So to make sure all of you understand how gel electrophoresis is actually done, like I'll show you actual instruments. Okay. Can we start, students, now? All of you drink some water. All of you drink some water and we'll start. Okay. Do not worry if you have not done this. I'll make sure you get the feel of it now. How much ever I can. Okay. Now, students, you might be wondering. The first question is why? Why gel electrophoresis? You might be wondering, why do we need to do the gel electrophoresis? Right? Remember, students, when we did the cutting of DNA, and we got the DNA fragments, do you think the DNA fragments are very small or is it very big? It is very small. DNA fragments are very, very small. So in order to find your desired DNA, we need to separate them out. Yes, we need to separate them out. And the separation of DNA based on the size is done with the help of gel electrophoresis. Now, is everyone ready to be a scientist now? Yes. All of you wear your coat. All of you wear your coat now. Yes, sir. I wore the coat now. Ready. Gel is your gel medium. Electro means related to electricity. Phoresis means being carried. So charge molecules, students. Charge molecules being carried in the electric field. Charge molecules are being carried in the electric field here. Now quickly tell me in the chat, is DNA positive or negative? All of you know DNA is negatively charged. Why? Because of the phosphate. Because of the phosphate here. Now, the first step in your gel electrophoresis, the first step is going to be making the agarose gel. Yes, we need to make our agarose gel. How do we make it? We take a agarose gel from your seedbed mixture. Now, students, quickly tell me in the chat, which algae do you use? Tell me in the chat, which algae do you use? Tell me in the chat. There is some algae which I'll tell you. I'll look at the chat. If I tell you, you will spoiler alert. It will become. You take your algae. Okay. Okay. And we make a agarose gel. We got our agarose gel. Okay. Then we make the gel matrix. With the help of your agarose gel, we make our gel. How do we do it? Agarose gel from the seaweed has pores which allows the DNA to move through it. That is called as your sieving effect. Sieving effect. With the help of sieving effect. This I'll tell you principle later on. Principle we'll see the later on. Principle I'll tell you later on once we're done with it. Okay. Now what do? Take the agarose gel, add it into a microwave. When we add into the microwave, what happens? The agarose gel will melt. It will nicely mix together. And it will melt. Now you take it out and we add something called as your ethidium bromide. Now why? Now why are we adding ethidium bromide? Because students, remember, DNA cannot be seen with a naked eye. No, you cannot see it. We need to add a dye called as your ethidium bromide is added. It helps in a visualization of your DNA. Next, what do? You take your agarose gel, we add it into a tray. Okay, done. Once you add into a tray, next we add something called as comb. Now once we add the comb and we take out the comb, can you see small gaps there? These small gaps are called as your wells. They're called as wells. Now, once we have the wells, can we add our cathode and anode? Yes, sir, we can add our cathode and anode. Okay. One more thing I forgot to mention here, students. Ethidium bromide will produce your orange color. Orange color. That is very important. Neet PYQ. That's why. No, Neet PYQ. Okay. Now, once we have the wells. Now, once we have the wells, we add our buffer here. Can you see buffer here? Buffer is there. We have our anode. We have the cathode. Okay. Now, friends, we take our DNA piece. We take our DNA plus DNA loading dye. Now, what is this DNA loading dye? Now, ethidium bromide is there to visualize the DNA after separation. Ethidium bromide is to visualize DNA after separation. But there is something called as loading dye. Now, why is loading dye important? Because if you do not add the loading dye, you cannot see if the DNA has moved, if the DNA has actually moved. And this loading dye can be seen with the help of naked eyes. It can be seen with the help of naked eyes, and loading dye is blue in color. Okay. Can you see this? Loading dye. This is how in the wells. This is how in the wells we add our loading dye. Okay. Loading dye plus the DNA. Loading dye plus the DNA. We have the DNA also here. Okay. So we add the DNA also here. DNA is done. Now what do? We add our electricity. Once we add electricity, what happens here? This is how the electricity looks like. We take our piece on top, we add here. Okay. Once we add, what happens? Once we add this, we switch on the machine. We switch on the machine. Switching on the machine, done. Starts. Electricity starts now. Electricity is starting. Electricity is running now. What happens when the electricity is running? Can you see the DNA is moving now? DNA is moving. Now let's come to principle. Can you see the dark blue color moving? Movement of dark blue color. That is your movement of DNA, which is visualized with the help of loading dye. With the help of loading dye, you can see the movement of DNA. Now let's go back to the principle. Let's go back to the principle of how this is moving. How this is moving. The main principle of gel electrophoresis is separation of different DNA molecules based on their size under the influence of electric field. Under the influence of electric field. Now see here very carefully. If you notice, DNA is negatively charged. It will move towards your anode. Yes, it will move towards the anode. As it is moving, larger DNA fragments will be left behind. Smaller DNA fragments will move further. Smaller will move further. Larger will be at the back. So principle of your gel electrophoresis, separation of DNA fragments based on the size. Based on the size. Okay, based on the size. Now students, once we have our separation, we do a visualization. We do a visualization. Now can you see ethidium bromide with your normal eyes, naked eyes? No. You need to take your this particular block of DNA, and visualize it under UV light. Visualize under UV light. So we take a UV trans illuminator. You take a UV trans illuminator, and then only you will find your separation of DNA. Now the one question will be like, sir, how do you know if this is a gene of interest? What if this could be a gene of interest, right? How do I know that? Simple. We have something called as your ladder. Where is ladder? We have something called as a ladder here. With the help of this DNA ladder, we know what is the size of our gene of interest. We know what is the size of gene of interest, right? We know on paper our DNA is around 2000 base pairs. Yes, and we can identify this gene of interest is around 2000 base pairs, and that's how we obtain our gene of interest. Next step is what? Next step is called as elution. Next step is called as what? Elution. In elution, what do? We take a particular suction force and we remove the entire gel block. We remove the entire gel block and we add it into a Eppendorf tube. We add it into a Eppendorf tube. Once we add the Eppendorf tube, then we give a heat block. Why? Because gel, agarose gel is very solid. We need to melt it little. Yes. Now we melt it. We have DNA. Now after melting, we add some amount of your chilled ethanol. Chilled ethanol is added. Once we add the chilled ethanol, DNA will precipitate out. Yes, DNA is precipitating out, and finally we have our gene of interest. And if our gene of interest was your insulin, and finally we have our insulin. Finally we have our insulin. Students, are you understanding? Are you understanding, students? This is how the entire process happens of your gel electrophoresis. And students, if you have any doubt, watch the video one more time in 1.5 speed. You will understand every single step of this process. Every single step. I could have just written down what happens in your gel electrophoresis. It is given in NCERT. But would you understand everything? No. When you visualize, this is how biology should be taught. Right? This, when you visualize and you learn, it's better. And I wish someday I could teach you on, like, you know, imagine if I had the instrument in front of me. If I could teach you with the help of instrument, it would have been much better. But since this is online mode, this is the best way I feel you can learn biology. Okay? Clear? Are you understand? Do you get a feel that as if you are doing the experiment? Right? Just imagine you are doing this experiment and you could have done it like this. Okay? Now let's take a quick example. Let's take a quick question. Let's take a quick question, students. Read the question, all of you. 2 DNA fragments are collected, A and B, assuming that A contains more base pairs than the B. So A has more base pairs than B. Select the incorrect statement with respect to gel electrophoresis if the DNA sample A and B are loaded in 2 wells. See, all of you know what are the 2 wells we saw. Answer the question now. Incorrect statement, all of you. Incorrect statement now. All of you is the incorrect statement. Students, if you're liking the session, if you're able to understand the concepts quickly, like the smash button. Smash the like button. Smash the like button, all of you. Smash the like button. Read the questions carefully. A will stay near the cathode than B. B will migrate with a greater speed compared to A towards the anode. A will migrate with greater speed compared to the B and the anode. Both A and B will form 2 distinct bands corresponding to their molecular weights. They are asking you, students, incorrect statement. They are asking you incorrect statement. I'll tell you quickly. Listen. If this is your gel block and this is the wells, if this is the wells, whose weight is more? Tell me, whose weight is more here? Obviously, students, here A has more weight. A has more weight. A has more weight. More base pairs, so more weight. So A has around 20 base pairs, imagine. Okay, let's take it 200 base pairs. A has around 200 base pairs. 200 base pairs. Your DNA B has around 20 base pairs. B has around 20 base pairs. Listen to me very carefully. A is around 200 base pairs. B is around 20 base pairs. That is assuming that A contains more base pair than B. A contains more base pair than B. Now in this particular, when you add A here, you add B here. Tell me which will go further, which will go faster. All of you know, if B's weight is less, according to the principles of gel electrophoresis, the lesser weight will go further. Yes. So B will be here somewhere and A will be here somewhere because A weight is higher. Okay. Now can you read the third statement here? A will migrate with greater speed compared to B towards the anode. This is your anode. This is your cathode. Can the A migrate with higher speed compared to B? Tell me now. Now, which is the correct answer now? Tell me. Select the incorrect statement. So, which is the incorrect statement now? Are you able to question? Are you able to understand, students? 1 second. I'll call someone now. Now, tell me the answer in the chat now. Tell me quickly. All of you know now? Yes. So this is your incorrect statement. That is incorrect statement. Now quickly solve this next question. Quickly solve this next question now, students. Are you understanding now? Are you understanding how the questions will be framed? If the concept is clear, then only you will be able to answer. If the concept is not clear, it gets difficult to solve questions. Bro, Mithun, can you come to the studio once again? Hello? Studio number 3, can you come? The students are saying the voice is a little low in studio number 3. Studio 3, yeah. Oh, chat went somewhere. It's live. It's live. They're saying voice is kind of low. Can you increase the volume? No, not here. Check, sound check. That is the maximum, right? Why are they saying?
Now ask.
Now you check, students. I'm Batman. Now students, check now the sound. Is it more louder? Yes, is it more louder, students?
Not this one. Let me change this as well.
Is it more louder now?
Is it okay?
Is it okay? They're complaining from that. They're crying from that time.
Oh. Now say yes.
Yes. Okay. Waiting to the rescue. Thank you. Sound check, students. Sound check. Is it more audible now? Is it better? Somehow better? The pranking me, it seems.
Now?
Sound check. Okay. Enough, I think that'll be enough, I guess, student. I think that'll be fine. Okay. Sound check.
I think this is enough.
Okay.
Okay.
Thank you, Mithun. This is your ethidium bromide. Next one is insertion of gene of interest into the vector. Vector DNA. Now can we start with the vector DNA, sir? Better now? Yes. Now, till now, any doubt in your gel electrophoresis? Till now, is there any doubt in your gel They're pranking me, oh my god. Till now, students. Now, till now, any doubt in your first segment of this chapter? Till now, there was any doubt in your gel electrophoresis, recession endonucleases, or it could be any other concept which we learned till now? No doubt? Yes, no doubt. Amazing. Now, can we start with your vector DNA now? Yes. Can we start with your vector DNA now? Amazing. Now, what is this vector DNA, students? A DNA sequence into which a gene of interest, this thing is clear now. Yeah. A DNA sequence into which gene of interest can be inserted is called as your vector. It acts as a carrier that carries the gene of interest into the host where it multiplies. Right? Where it multiplies. All of you know vector. We have been using vector from that time. Example is your plasmid. Now, what is the role of vector? There are two main roles of a vector. The first one is going to be carry the gene of interest. It is taking the gene of interest and it is carrying into the host. The other one is going to be your producing copies. Now, what are the different types of cloning vectors? The first one is going to be a plasmid. Right? Now, what is then we have phages, then we have back, then we have yak. Now, what are the different what is a plasmid? What is a phage? What is a back? What is a yak? Listen to me very carefully now, students. Listen to me very carefully. Now, what is a plasmid? Plasmid is nothing but extra chromosomal DNA, an extra DNA present in a cell other than the genetic material. It is extra self-replicating circular double-stranded DNA, circular DNA present in a bacteria and some yeast as well. That is your plasmid. Then we have bacteriophages, virus that can impact bacteria and bacteriophages. It consists of your linear DNA, which can also be used as a cloning vector. Then we have back and yak. Now, what is back? Bacterial artificial chromosome derived from your bacterial DNA. Then we have yak, that is yeast artificial chromosome derived from your yeast. Both are genetically engineered. They are used in your human genome project. So if they ask you a question, which particular vector was used in your human genome project, the answer should be your back and yak. Now, question for you. Can any random DNA sequence be a cloning vector? You are telling me in the chat. Can any random sequence, right? Can any random DNA sequence be a cloning vector? Or are there any specifications? Yes. Tell me in the chat. Can any random DNA sequence become cloning vector? Or do we need to have a specific requirements? Do we need a specific requirements for your cloning vector? Anyone? Yes. There are specific traits for an ideal cloning vector. Now, what are the specific traits here? Listen to me very carefully now. It should have ability to self-replicate. That is, it should have a ori site. Right? Ori site, that is origin of replication, should be there. Then it should have a repressor of protein, that is ROP site. It should have a selectable marker. It should also have a cloning site. Selectable marker to differentiate between transformant and non-transformant. Then we should have a cloning site. Cloning site is a site in which our particular restriction enzyme can come and cut. Okay, come and cut. Now, students, which is the most ideal one which is given in your NCERT? The most ideal one which is given in your NCERT is your PBR 322. PBR 322. Now, what is the meaning of PBR 322? That is plasmid, discovery, Bolivar and Rodriguez, and 322 is your order of discovery. That is plasmid, Bolivar, Rodriguez, as well as your order of discovery. Now, students, now can we start one by one? Decoding this particular diagram which is given in NCERT? Because a lot of questions can come from this diagram. This particular diagram here. Okay, listen to me very carefully. This is your, first of all, E. coli cloning vector. It is a E. coli cloning vector called as your PBR 322. Now, plasmid, Bolivar, Rodriguez, order of discovery 322. It has two different selectable markers. Now, what are these selectable markers, students? Selectable markers are your markers which will help you to distinguish, identify which is transformant, which is non-transformant. Okay. Here we have ampicillin resistance gene. Can you see R here? R written here means it is resistance. Ampicillin resistance and tetracycline resistance. In ampicillin resistance, we have your PST, PU1, EcoR1. These are your cloning sites. They are the cloning sites in which a restriction enzyme can come and cut. These are locations where restriction enzyme can come and cut here. Then we have your Cal1 and Hind3. Then we on your tetracycline we have your BAMH1. Now, is this important to remember? Yes, it is very important to remember. Very, very important. Then we also have your Sal1 on your tetracycline. You need to know on tetracycline we have BAM1 and Sal1. You should also know on your ampicillin we have PST1, PU1, EcoR1. Okay, EcoR1 is here, sorry. E carbon is here. We have your PU1 and PST1. That is very important. Then we have origin of replication, ori site. Then we also have ROP, repressor of protein. Repressor of protein. Okay. On ROP we have your PVU2. PUV2. That is, origin of replication is a sequence that initiates DNA replication. It is initiating DNA replication. An ideal vector should have an ori site. This property allows the vector to multiply within the host. Now I'll ask you a question. If there is no ori site, students, if there is no ori site, can the vector multiply inside the host? No. Remember in the starting we took a particular vector from your Pandu. When we take the vector, when we have the vector recombinant DNA, right, recombinant DNA, when we add it into the matcha, right, when we add it into the matcha, can the replication happen if there is no ori site? No. So ori site is very important for it to multiply. For it to multiply. Yes. Now it should have a repressor of primer or repressor of protein. That ROP gene codes for your ROP proteins. That is, these proteins are involved in replication of vector. Replication of vector. Done. Now it should have a selectable marker. Now what is a selectable marker? Selectable markers are genes that help in identifying the rDNA is if recombinant DNA has successfully entered inside the host, inserted into the host. There are two types of selectable markers. One is called as your antibiotic resistance genes. The other one is called as your laxant genes. Now working of these two, working is very important, and as well as the principle is very important. Working as well as the principle is very important, which I'll be teaching you in next 5 minutes or 10 minutes. In next 10 minutes or 5 minutes, I'll be teaching you that. And students, if you remember that, if you learn selectable marker, if you learn what is laxant gene, how does it function, what is insertional inactivation, this chapter is done for you. This chapter is done for you then. Okay. Now students, in PBR 22, antibiotic resistance genes is there. That is, ampicillin resistance gene and also tetracycline resistance gene is also there. Okay. It should have cloning sites. Does it have cloning sites? Yes, it has cloning sites. Do you see cloning sites here? Yes. We see multiple cloning sites here. Yes. That is the site at which gene of interest is inserted. That is, it is a site, cloning site is a site in which your restriction enzyme will come and will cut there. It will come and cut there, make a cut, and we add a gene of interest here. We add a gene of interest in this region. We add a gene of interest in this region. Okay, gene of interest in that particular region. Using that is, it will have restriction enzyme site or recognition sites will be there. Okay, so if the recognition site is there, it will come and cut there. It will come and cut there. Then using DNA ligase enzyme, the gene of interest is ligated later on. Students, did I tell you working of restriction enzymes? How does it work? Did I tell you? Did I tell you how does it work? I don't remember. I forgot almost. I think I told you, right? How does the restriction enzyme work? I told you, right? Or do you want a small story on that also? Did I tell you how does the restriction enzyme work? How does it cut the DNA? I told you, right? Yes, I told. Now remember students, I'll tell you a small story how to remember it also. I'll tell you a small story how to remember this. Okay. Quickly tell me in the chat, which is your favorite biscuit? Quickly tell me in the chat, which is your favorite biscuit? Everyone in the chat, which is your favorite biscuit? Yes, I'll give you a break after this. Which is your favorite biscuit? Which is your favorite biscuit? Tell me first. Which is your favorite biscuit? Tell no, which is your favorite biscuit? All of you are very shy, huh? You won't tell. Okay, I'll take mine only favorite. I'll take my favorite biscuit. Huh? Okay, mom's magic. Priya is saying Oreo. Okay, let's take Priya's example today. Okay, hide and seek is my favorite. Hide and seek is also my favorite. Okay, hide and seek is also my favorite. Okay, let's take Priya's example. Priya is saying Oreo. Okay, now imagine Priya is going into the supermarket. Imagine she's going to Reliance Fresh. She's entering Reliance Fresh. Priya has entered the Reliance Fresh or market. She will look at all the biscuits. Oreo is here. Okay, I saw the Oreo. Dark Fantasy I saw. I saw your Parle-G biscuit. I didn't see anyone say Parle-G. Bourbon. Unibake. Mama's Magic. Priya will do what first? She will go look at all the biscuits. She will go look at all the biscuits. And then she will finally come where? After looking all the biscuits, she will come back to the Oreo only. Yes. She will take up the Oreo. Yes, she'll take up the Oreo. Your restriction enzyme also works like that. Restriction enzyme also works like that. If this is your piece of DNA, students, if this is your piece of DNA, restriction enzyme will read the entire DNA. Restriction enzyme will look at the entire DNA. It will inspect the DNA. Yes. When it finds the recognition sequence, it will go and bind there. It will go and bind there, and then it will cut. After cutting, what are we left with? After cutting, we are left with your hanging ends. Hanging ends or overhang ends. Overhang ends. Just small story. Okay. Just milk bikkies. Yeah, milk bikkies is memory. Now, students, that was your entire PBR 322. Does anyone has any doubt in PBR 322? PBR 322. You should know what is your ORI site, what is ROP, what is the meaning of PBR 322, what is the meaning of AMPR, what is tetracycline R, what is the cloning site. All of you know what is a cloning site? Cloning site is a site. Cloning site is a site which helps in recognize, which has your where a gene of interest will add, where your gene of interest will come and attach, and it also has your recognition sequences. It also has your recognition sequence. Recognition sequence is also there. Okay, done. Jimjam. Oh, Jimjam is also super good. Nancy is saying Parle-G. PUBG? No. Parle-G. Yes, Parle-G and tea. Nothing can beat it still. Parle-G and tea. Nothing can beat it still. Okay. Now let's understand something called as TI plasmid. Let's understand something called as TI plasmid. Ready? Ready, students? Why everyone has become so quiet now suddenly? Any trick to remember? Pilgrim is asking, any trick to remember this? I have to make a trick. Can I make a trick and upload a video on this, short on this? Can I don't have time to think now. I'll think later. It takes some time to think. I will think and I'll make a short on this. Okay, how to remember your all of this and all of this. Okay, works. Okay, works. Now, students, let's understand your TI plasmid now. Okay, let's understand TI plasmid. All of you drink some water now. Rehydration is very important. Listen to me very carefully now. I want your ultimate attention, students. I want your ultimate attention now. You learn this concept one, you learn this concept, and you learn one more concept called as your insertional inactivation. This chapter is done. This chapter is done then. PCR is very easy. PCR is very easy. And last concept called as your bioreactors is also very easy. Okay, bioreactors also very easy. Okay. Now listen to me very, very carefully now. Listen to me very carefully. Now we have something called as your Agrobacterium tumefaciens. Agrobacterium tumefaciens is your bacteria. This Agrobacterium tumefaciens typically infects your dicot plants. It typically infects your dicot plants. It typically infects your dicot plants. Okay, dicot plants. Now this particular Agrobacterium tumefaciens has something called as your, this is something called as your, TDN is already written here. Okay, let us TI plasmid. TI plasmid. Also called as your tumor inducing plasmid. Tumor inducing plasmid. Okay, this is your tumor inducing plasmid. This tumor inducing plasmid also has something called as your tDNA region. tDNA region. Why? This is called tDNA because it is also called as your transfer DNA. It's also called as what? Transfer DNA. Okay, it's also called as your transfer DNA. Now this transfer DNA, once it is added to your plant cell, what happens? Now if this Agrobacterium tumefaciens, if it infects a dicot plant, if this Agrobacterium tumefaciens, if it infects a dicot plant, plant cell, this tDNA, this transfer DNA will attach, will it enter, once it enters the plant cell, it will join your genome of your plant. It will join the genome of your plant. Now, once it attaches genome of plant, there is increase in a hormone called as your cytokinins. Increase in cytokinins. There is increase in auxins also. Increase in auxins and cytokinins. If there is increase in auxins and cytokinins, can I tell there is increase in growth? Yes. There is increase in growth. Uncontrollable growth. If there is uncontrolled growth, it results in a disease called as your crown gall disease. 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 Okay. Now, scientists identified that this tDNA region is harming the plants, but without any, it is harming the plants. So, what do you do now? What do you do? You basically take this tDNA and delete it. You take the tDNA and you completely delete it. Once you delete the tDNA and you add your desirable gene instead of it, instead of this tDNA, you add your desirable gene. Desirable gene. Okay. We are adding a desirable gene. Now, now the desirable gene is also entering inside. It is also able to produce. Once entering the, if a desirable gene is entering inside, can I say your desirable gene is being produced here? Yes, it is being produced here. So we are using a simple method. We are using a simple method of your tumor inducing plasmid to infect your entire plant with your desirable gene. Inside your plant with desirable gene. See here, agrobacterium tumefaciens, TI plasmid. It is entering your plant chromosome DNA. It is fixing tDNA, joining there, and it is producing a crown gall disease. But instead of that, instead of your crown gall disease, we can take our desirable gene, and with the help of this process, we can insert our desirable gene into the plant and make the plant produce our desirable product. Make a plant produce our desirable product. Understood? Tell me that quickly. Did you understand that? Or should I repeat? Did you understand? This is happening in your plant cells. This is happening in your plant cells. See, this is a natural process. Basically, what are we doing? We are taking the tDNA region. We are removing the tDNA region. And we are adding our alien DNA. Alien DNA or your desirable DNA. Desirable DNA, and with the help of desirable DNA as well as your alien DNA, we are infecting the plant and it is able to replicate inside. Okay. Now, next thing. The next concept is very important. Insertion of DNA into the bacteria. Insertion of DNA into the bacteria. Now listen to me very carefully, students. Listen to me very, very carefully. We have a simple living DNA here. Simple living bacteria. We have a simple Papa bacteria here. Very Papa, very innocent bacteria. Okay, very innocent bacteria. We take our rDNA. What is rDNA? Your recombinant DNA. We take a recombinant DNA, and if we take this recombinant DNA and we make it enter into the bacteria, will the bacteria keep quiet and take the rDNA, or will it fight against it? Tell me in the chat. Obviously, students, obviously, this bacteria will fight against it. It will be like, no, why should I take it? It will be like, why should I take your DNA? Why should I take a separate extra rDNA? It will fight. How will it fight? If you look at the membrane, here and the membrane of your recombinant DNA, right? They are negatively charged. Moreover, bacterial cells have hydrophobic membrane while your DNA is hydrophilic. They will have some kind of, dissimilarities. They'll fight against it? Yes. So how do we make it take it up? How can we resolve this fight? We can resolve this fight with the help of a divalent cation, such as your calcium. So we take a divalent cation, such as calcium, and which makes the membrane difference. Yes. Now once we add divalent cation, the easily our recombinant DNA can enter into the bacteria. Okay, once we add a divalent cation, this calcium will make small holes there, small pores. Divalent cations such as calcium ions, small pores in your bacterial cell wall, and it can, our recombinant DNA can enter inside. Okay, recombinant DNA can enter inside. Now such bacterial cells which can take the DNA, they are called as your competent cells. They are called as what? Competent cells. Cells that are capable of taking up the DNA from external are known as your competent cells. And how do you make the competent cells? With the help of divalent cations. And that process, the process of transfer of your exogenous naked DNA, also called as your alien DNA, into the host is called as your transformation. It is called as transformation. We have multiple methods. The first method is called as your heat shock method. Then we have micro injection. Then we have ballistics. Then we have your disarmed pathogen method. Okay, disarmed pathogen we already studied. That is your TI plasmid. This is your TI plasmid method. Now we'll study the heat shock method. Now what do we do in heat shock method? Very simple. We'll shock the bacteria. We'll not give the bacteria to take its time to react only. We will completely shock it. How do you shock it? See, imagine we have the competent bacterial cells. We are taking competent bacterial cells. Remember, these competent bacterial cells can take up the DNA because they have been treated with your calcium ions. Yes. And we take your rDNA with your gene of interest. Now we add it here. Once we add it here into a Eppendorf tube. Once we add an Eppendorf tube, you take it into the ice. You take the tube and we add into the ice. Then you take into the heat, again add into the ice. I'll repeat. Take this tube, add it into the ice. Suddenly you take it out and you keep it in a heat, like boiling water. Keep it in boiling water. Again take it back and keep it into the ice again. Bacteria is shocked. Your gene of interest rock. Yes, the bacteria was shocked. You give it a heat shock so that the bacteria doesn't react. Bacteria is not reacting now. So competent bacterial cells with rDNA is now placed in your water bath temperature at 4 degrees Celsius. Again keeping back in the ice. Back to the ice. Now the transformed bacteria cells are now plated on and then incubated at the room temperature. And once we check inside the microscope, under the microscope, we know rDNA directly. Oh, this is a different one. When we check under the microscope, we realize that your entire rDNA has entered inside the bacterial cell. That is the first method. Second method is called as your micro injection. What do? You take your rDNA in an injection and you directly inside the bacteria. I'll show you diagrams here. See, this is your bacteria. You take an injection and directly inside you insert your vector. You insert your rDNA. rDNA directly inside the bacteria. That is called microinjection. Then we have ballistics. Ballistics is like your gun. Gene gun or ballistics is very simple. In gene gun or ballistics, we are taking. Okay, I'll draw and show you. See, in gene gun is very simple. In gene gun, if this is a plant and this is the leaf, you take a gun, small gun you take. You take a gun and directly with the help of this gun, you can add your gene into the plant cell, directly into the plant cell. But there is one more method. There's one more method called as ballistics. In gene gun, there's something called as ballistic also. That I'll show you. In ballistic method, what do we do? If this is your setup, this is your ballistic method. You take high velocity particles. You take high velocity microparticles. Example like your gold and tungsten coated with DNA. So we have high velocity particles coated with your DNA. When the high velocity particles are coming like this, and once they collide here, what happens? The entire DNA will be sent to your host. Entire DNA will be transferred to the host, but all the high velocity particles will be left here. High velocity particles will be left here. So what do we have? All the DNA will enter the host. All the DNA will be entering into the host now. DNA in host. DNA in host. Clear? Gold and tungsten, that is very important. It is the P by Q. Okay. All of you know the tumor inducing. We learned already. Crown gall disease. Okay. Crown gall disease. Now, the next one is selection of your, next concept is your selection of your recombinant colonies. Selection of recombinant colonies. Okay. Now students, if you have understood till now, quickly smash that like button. And I want to see some Josh now, because the next concept is very important, and if you pay your attention for the next 10 minutes, students, for the next 10 minutes, I want ultimate attention. Can you give me? For the next 10 minutes, I want ultimate attention. Can you give me that? Tell me in the chat. I'll look at the chat here because chat is a little lagging there. For the next 10 minutes, students, I want ultimate attention. Can you give? Next 10 minutes, just 10 minutes. Ultimate attention. Yes. And do you have any doubt? Do you have any doubt till now? Ask me right now in ballistics or gene gun or micro injection, or could be your heat shock method. I believe it was very clear, right? I'm deliberately going a little slow here so you can understand this concept and not make a mistake here. Okay. 10 minutes, ultimate focus. Because one question will come from here. Okay. Listen to me very carefully now. Listen to me very carefully. We will learn a concept called as your selectable marker now. We'll learn a concept called as your selectable marker. Now, what is this selectable marker? What is this selectable marker? Listen to me very carefully now. Do not worry. If you don't understand, ask me again, but learn this concept very clearly. Okay. Now, what is selectable marker? Students, if you notice here, this is ampicillin R. This is tetracycline R. Now, if I take this PBr 322 and add it inside ampicillin, or if I add it inside tetracycline, will it survive? Yes, because it has genes for ampicillin, it has genes for tetracycline. Okay, cool, sir. Now, students, we take our gene of interest. Now we have a gene of interest. All of you know gene of interest. How do we obtain gene of interest? All of you know. By cutting a gene of interest, we have a gene of interest now. We take this gene of interest and we want to add it into a vector. Gene of interest into a vector. Now, how can we add it? We also need to make a cut here, right? We also need to make a cut here. See, if this is a segment here, if a gene of interest is a segment like this, this is a gene of interest segment. This is a gene of interest segment is there like this. How can we add it into this? Unless we cut here, then only we can add this. Yes, then only we can add this. So imagine our restriction enzyme. Our restriction enzyme is going to be your BAMH1. Restriction enzyme is going to be BAMH1. So your restriction enzyme will come and cut here. It will come and cut here. Okay, it's cutting. So what do we allow? What do we have now? This is your vector. In this vector region, can I say, if we remove this part, and add our gene of interest, this tetracycline resistance gene is lost. Yes, this tetracycline resistance gene is lost now. So whatever we have read right now, whatever new vector we have right now, can I say it is tetracycline sensitive? Can I tell? It is tetracycline sensitive now. Yes, it is tetracycline sensitive now. Yes. Now we take this vector and we add it into the host. This is your host cell. This is the host. We take the tetracycline and we add it into the host now. That is called as what? That is called as your transformation. Yes, it is called as what? Transformation. And how do we do transformation? Gene gun, heat shock method, ballistic method. Yes, all of these are different. Micro injection. All of these are methods are of your transformation. Now students, your entire vector is inside your host. Vector is inside the host. Okay, it is inside the host. Three possibilities here. there are three possibilities. Yes, there are three possibilities here. What are three possibilities? This is a host. We have three hosts here. We have three different hosts here. Okay, three different hosts. Okay, now what if in the host A, let's call it A, B, and C. What if in the host the recombinant DNA did not even enter? There's a possibility that, right? There is a possibility that in your host A, there's a possibility that the vector did not enter itself. Yes, that's a possibility. Vector did not enter itself. So can I call it non-transformant? Yes, it can be called as non-transformant. Now students, in the next host, is there a possibility that whatever vector enter inside, it did not have our rDNA, right? It did not have our rDNA. Yes, it cannot have your rDNA. Yes, it cannot have rDNA. So can I call it non-recombinant? Yes, it is your non-recombinant, because non-recombinant does not have our rDNA. Does not have rDNA. Yes. Now what about the third one? In the third host, do you see our rDNA? Yes. Do you see our rDNA? Yes. We see our rDNA here. We see our rDNA here. So can I call it recombinant? Yes. We can call it recombinant. So students, let me summarize again. Starting, we had your PBr 322 vector. To this vector, we added our gene of interest. Now, once we add a gene of interest into this vector at tetracycline region, this particular segment has been removed out. Once we remove this segment out and once we add our gene of interest here, tetracycline resistance is lost. Tetracycline resistance is completely lost here. So this becomes tetracycline sensitive. Yes. Then we had three possibilities that your host, once we add into the host, when you do the transformation, possibility one is non-transformant. That is, our vector did not enter at all. Our vector did not enter at all. What about the second case? In second case, there is non-recombinant. That is, vector is there. Vector is inside. This is your vector. Vector is inside, but it does not have rDNA. rDNA is missing here. rDNA is missing. But in the third case, students, in the third case, rDNA is present. Yes, rDNA is present. Vector is also present. Vector is also present. Now, quickly draw a table now. Quickly draw a table. Yes, let's draw a table. Yes, let's draw a table. This is A, this is B, and this is C. This is A, this is B, C. Now we take all the three. We take all the three hosts and we add it into a ampicillin media. A media containing ampicillin. Yes. We add into a media called as into a ampicillin. Now, you tell me, can the A survive in ampicillin? Can it survive in ampicillin? No. Why, sir? Why it cannot survive in ampicillin? Because it doesn't even have the vector. If it doesn't have the vector, does it have ampicillin resistance gene? Does it have tetracycline resistance gene? No. It doesn't have both of them. So can it survive here? No. When you add your host A in ampicillin, it cannot survive. It does not have any type of resistance gene. Yes, it cannot survive. Now what about B? Yes, B can survive because it has the vector and the vector has your ampicillin resistance. It can survive. Now what about C? Can it survive? Yes, it can also survive because it also has your ampicillin. It also has your ampicillin. It can survive. Okay. Now you take this same three hosts. Now you add it into tetracycline. You add it into tetracycline. You add it into tetracycline. Okay, tetracycline. Once you add into tetracycline, can this survive again in tetracycline? You will say no, sir. It cannot survive in tetracycline because it does not have vector, and if there is no vector, there is no resistance. Yes, No, not survive. Can this survive? Yes, because it has your tetracycline also. It has the tetracycline, it can survive easily. But can this survive? No, it cannot survive, because as soon as we added a gene of interest, this particular segment became inactivated. It became what? Inactivated. That is called as insertional inactivation. So our gene of interest, that is, which is present in your C, our gene of interest which is present in your C will not be able to survive in your tetracycline. Will not be able to tetracycline. That's how we obtain your transformant, non-transformant, non-recombinant, as well as your recombinant. Now you will have one question in your mind, all of your mind. Sir, we wanted C. We wanted our gene of interest was present on C, but you killed it. What is the experiment now? Is the experiment fail? You might be thinking, right? Sir, our gene of interest is present in C, but you killed it. You killed it completely. How will we even do the experiment now? What is the use of doing all of this? You might be thinking, right? Students, remember, when we are doing any of these experiments, there's always triplicates. There's always triplicates are there. We make multiple replicas, we do it. Even though in first batch we killed everything. In first batch we killed our desirable. But we got to know if it is recombinant, right? We got to know if it is recombinant. We'll use the other two. We'll use, if the batch 1 is used for testing, batch 2 will be used for next step. Batch 2 will be used for next step. Batch 2 will be used for next step. Okay, batch 2 will be used for next step. First step is the first batch. First batch is used for identification. Second batch and third batch or fourth batch can be used for next steps. Understanding? Every single thing is in triplicates or quadruplets to make sure the experiment is maintained throughout. Okay, so with the help of this is called as insertional inactivation. Now students, do you notice this is a very difficult process? That is, you need to add your insertional inactivation, then you make triplicates and then you do it. Can I call it a cumbersome process? Yes, it is a very difficult process. Too much of work is going in this. We want some shortcut method now. Like how you write. See, all of you are saying, Reddy is saying KK, sir. Reddy is saying KK. Instead of saying OK, he is saying KK. Yes. Then we have some students will be like, yes sir, happy birthday. Instead of saying happy birthday, you write HBD. All shortcut you want. Yes. So can I tell you a shortcut method? Can I tell you one shortcut method now? I'll take the shortcut method. Listen to me very carefully. The shortcut method is even very, very much simple. In shortcut method, we use a different plasmid. In shortcut method, we use a different plasmid only. The plasmid method is, let's see. The other plasmid is called as your PUC8. We have a plasmid called as your PUC8. We have PUC8 here. Now this PUC8 has two regions. This PUC8 has a amphicellular region here. It has a amphicellular region here. Yes, it has a amphicellular region here. It has a separate region called as lag Z region. In this lag Z region, we have the lag Z gene. In this lag Z region, we have the lag Z gene. Okay. Now all of you know molecular base of inheritance, which you just learned. Now quickly tell me in the chat, lag Z gene is responsible for which particular substrate production? Tell me in the chat. All of you know, right? Lag Z gene is responsible for production of your beta galactosidase. Yes, beta galactosidase. Yes, beta galactosidase. Now to this beta galactosidase, friends, to this beta galactosidase, if we add a chromogenic substance to this beta galactosidase, if we add a chromogenic substance called as X-gal, called as what? X-gal. X-gal. We add something called as X-gal. This particular colonies, your entire colony of bacteria of this particular vector will look blue in color. It will look blue in color. Okay, students, I'll repeat again. We have a plasmid here called as PUC8. It has amphicellular region. It also has your lag Z region. And in this lag Z region is responsible for beta galactosidase. To this beta galactosidase, if we add X-gal, it produces a blue color substance. Blue color it produces. Now listen to me very carefully. Listen to me very carefully. Listen to me very carefully. We have our plasmid here again. Yes, we have a plasmid here again. And this is your lag Z gene. Yes, this is your lag Z gene is there. This is your lag Z gene. Okay, this is lag Z gene. Now students, listen to me very carefully. Listen to me very, very carefully. To this lag Z gene, I am cutting this segment. I'm cutting this segment and I'm adding my gene of interest here. I'm adding my gene of interest in the lag Z gene. Yes, I'm adding my gene of interest in the lag Z gene. Now tell me what happens now. Tell me what happens. Tell me quickly. Once we add our gene of interest or your alien DNA into the lag Z gene. Now will the lag Z gene, yes, will the lag Z gene still produce beta galactosidase? Tell me in the chat quickly. Will it produce beta galactosidase? Lag Z gene is also, remember, lag Z gene is only not there now. If there is no lag Z gene only now, beta galactosidase is also not there. Yes, beta galactosidase is also not there. So when we add X-gal again, a chromosome substance, if there is no beta galactosidase, do we get blue color? No. Do we get blue color? No. What do we obtain? We obtain now white color. We obtain white color now. We obtain white color. Now, what do we understand from this experiment now? What did we understand, students? That is the most takeaway. The biggest takeaway here is going to be this. The biggest takeaway is going to be here. I'll tell you. Biggest takeaway is going to be here. In this type of insertional inactivation. See, did we insert something here? Yes. Did we inactivate this lag Z gene? Yes. As soon as we added our gene of interest, lag Z gene became completely inactive. Yes. So when we have in these experiments, recombinants will look white color. Yes. Recombinants, when we added a gene of interest, it became recombinant, right? When we add a gene of interest, it became recombinant. So recombinant will look white color and non-recombinant here, when our gene of interest is not there. This is normal. This is non-recombinant. Gene of interest is not there. So non-recombinant will look like what color? Non-recombinant will look like blue color. Will look blue color. Color? Blue color. That is, whenever we do not add when we don't add a gene of interest, it looks blue color. But as soon as we add a gene of interest, we are inactivating a particular segment of lag Z gene and we are getting now white color. This is called as your insertional inactivation. As soon as you insert something, it is becoming inactivated. Clear? And this is also called as your blue and white screening. It's also called as what? Blue and white screening. Clear? Blue and white screening. Where's my phone? Also called as your blue and white screening, students. Did you understand? If you understood, quickly like the video and smash that like button, and little blue hearts, green hearts in the chat if I have to know. Green hearts in the chat if you understood. Now here, let's quickly summarize the entire concept which we learned right now. That is antibiotic resistance gene. We learned, right? Antibiotic resistance genes. What happens in antibiotic resistance genes? This is your PPS 22. We take our gene of interest. We cut this part here. In antibiotic, we cut this part. We remove this part. Yes. And we take a gene of interest and we add it here. Yes. Then what do I say? Tetracycline resistance has been removed. Only ampicillin is left. This particular PBR 322, it is now tetracycline sensitive. It is now tetracycline sensitive. Yes, that's what we learned here. Clue. Then this is a combustion process. Then we have the example is PUC8. In PUC8, can you see it has a gene called as your LAGZ gene. We have ampicillin gene. Now what do we do? If the LAGZ gene is present, it is producing beta glucosidase. It is producing blue colonies. Can you see under your, if you look at your colonies, if you look at the colonies, it will look completely blue color. But as soon as we remove this, gene of interest is coming. Gene of interest is coming, making a cut here. We are taking a gene of interest, we are adding it here. Yes, gene of interest is added here. As soon as we have gene of interest, LAGZ gene becomes completely inactivated. Yes, if there is no LAGZ gene, right? If there is no LAGZ gene, there is no beta glucosidase. If there is no beta glucosidase, can we get white colonies? Yes. Do you see some white colonies here? Yes. So how do you now take up your white colonies, students? Now what do you do in the next experiment is, you carefully take only the white now. Carefully, just take the white, leave the blue color. Like, don't take the blue color. Done. Just take the white color and do your experiment. Do your experiment. That's it. Did you understand? That is your blue and white screening. That is your blue and white screening. Understood everyone, Sanjana? Reddy, Pilgrim, Meghana, Satya, Supriya. Yes? Sanvi? Now, can we go to the end of the chapter now? Can we go to the end of the chapter now? Yes, sir. Students, in 2.5 hours, your almost chapter is over. Now we are going towards the end of the chapter now. Can we start the end of the chapter? That is the process of rDNA technology. The process of rDNA technology. What is the end of it? That is, look at all the steps here. Look at all the steps. We have first step is isolation of genetic material. Did we learn that? Yes, isolation of genetic material. Then we have cutting off DNA at specific location using your restriction endonucleases. Yes. Then we have amplification of gene of interest using PCR. That is the third step. Then we have insertion of your recombinant DNA into the host. Transformation. No, this is not transformation. This is insertion. This is just insertion. Okay. Yeah. Insertion of recombinant DNA into the host. Transformation. Exactly. Transformation. Then we have obtaining the foreign gene product. Then we have downstream processing. That is your fifth as well as sixth. Fifth as sixth. Now students, if you have understood till now, I want everyone to like the video right now and subscribe to the channel. And also students, if you're understanding these concepts, okay, quickly let me know in the comment section in today's video if you're able to understand the killer visualization. Okay, all the killer visualization. And also if you want to join my Telegram channel, you know how to find it. Okay, because on my Telegram channel I will be sending you more and more images. Like when I did my electrophoresis, when I did the entire genetic engineering, those images I will be sending on today. I'll be sending on this. And if you want more questions, students, if you want more questions on this engineering, I will send a DPP also on a Telegram today. Okay, on DPP also. Now students, the first step. What is the first step here? The first step is isolation of genetic material. Isolation. I'm saying isolation. Isolation of genetic material. Now, how do you isolate genetic material, students? How do you isolate genetic material? Very simple. Very simple. That is going to be your, if this is your cell membrane, students, if this is your cell membrane, we have to break the cell membrane. Yes, you have to break the cell membrane. Now, how do you break the cell membrane, students? In the case of your bacteria, it has bacterial cell wall. Yes, you use lysozymes. You use lysozymes to break the entire cell wall here. Okay. Now imagine in the case of plants, we have the cellulase. We have the cellulase. Done. Broken down. Then we have, in the case of fungus, we have chitinase, which will break the membrane here. Yes. Done. So first step is done here. First step is done. Okay. Now what is the next step? Listen to me very carefully now, students. First step, cell wall is broken. What about your cell membrane, plasma membrane? Plasma membrane can be dissolved with the help of detergent. With the help of detergent, we can completely break the cell membrane or plasma membrane, and all the lipids will completely disintegrate. Lipids will completely disintegrate. Okay, that is the second step. Now, third step, students. Now, in third step, what happens? We need to make the protein disappear. How will you make the protein disappear? Tell me in the chat quickly, everyone. Tell me in the chat quickly, how will you make the protein disappear now? Everyone, quick. I want quick answers now. Yes. Everyone? Protein can be disappear with the help of proteinases. Yes. Proteinases can be added and we can disappear a protein. How will you disappear RNA? If you add RNases, RNA will also disappear. Done. RNA is also disappeared now. Now what are we left with now, students? We are left with your nucleases. We had your nucleus. We are left with nucleus. Okay. Left with nucleus. Now should we add nucleases now? Should we add nucleases to this? You will be like, oh, add sir, what will happen? Entire DNA will break down if you add nucleases. So you do not add nucleases, you take the DNA. You take the DNA now. Okay, you left with the DNA. Now students, if you want to precipitate the DNA, you add chilled ethanol. What do you add? You add chilled ethanol. Once you add the chilled ethanol, then you can see the DNA will start to precipitate. Once you add the chilled ethanol, the DNA will start to precipitate. Now once the DNA is precipitating, now you have to do something called as spooling. We'll do something called as spooling. Do you see this? We add a straw and we take out the DNA out. We take the DNA out. That is called as spooling. Okay. But is there any other easier method? Yes. That is your centrifugation. Take chilled ethanol plus cell extract. Add centrifuge and you will have pellets of your pure DNA supernated with cell debris. Remove the supernatant, take the pellet which has your DNA, Done. Now once you have your DNA, we need to cut the DNA. How do you cut the DNA? Everyone tell me, chat. Cutting of DNA at specific location using restriction enzymes. Done. Restriction enzymes have been done. You cut the DNA with the help of restriction enzymes. Now once you make small fragments, you need to separate the small fragments. How do you separate small fragments? With the help of gel electrophoresis. Second step. With the help of gel electrophoresis, you separate the bands. Done. Now once you have the separate bands, of your DNA, in your experiment I told you, you need to make multiple copies. Now how do you make multiple copies? Can anyone tell me in the chat? With the help of a process called as your PCR. And who discovered PCR? The PCR was discovered by Carimulus. Inventor of your PCR. Now what does PCR stands for? PCR stands for your polymerase chain reaction. That is a process of selectively amplification of your specific regions of your DNA in vitro multiplication. Okay, in vitro multiplication. Now students, are you ready for PCR now? Tell me in the chat quickly. Are you ready for PCR? Yes sir, we are ready for PCR now. Now do you want to be a scientist again, or do you want to be a scientist again, or do you want me to tell the principle directly? Tell me, do you want to be a scientist again and do you want to do the experiments, or do you want to be a do I directly tell you? Yes. Want to be a scientist again? Tell me in the chat quickly. If you want to be a scientist, I'll tell you exactly how PCR has been done. Okay, because I remember when we were in MSc, we used to do the PCR. And PCR used to take around 4 or 5 hours, and we used to go out. Completely. See, PCR used to be here every day. Come in the morning, take your DNA segment, add into the PCR, and then just go out. That was the best time. Doing PCR was the best time, students. When you do the PCR on your own, you will realize. Okay. So all of you, wear your coats. Now. Done. Scientist code is done. Okay. Now students, what do you need to do? First thing you need to know is what is the difference between what you need to know is what is the difference between in vitro and in vivo. In vitro means outside the living cell. What is the in vivo? In vivo is within the cell. And remember students, entire PCR is done inside a machine called as your thermocycler. It is called as what? It is called as thermocycler. See, this is the machine. It will look like this. So you need to press the button here. Can you see the buttons here? Press the button here. Select the number of cycles, and this lid will open here. And you need to add your DNA there. You need to add your DNA there. That is thermocycler, machine that allows the multiple replication cycles of a particular fragment of gene of interest. Now students, what do you need to add? Wait. Tejaswini, everyone, Supriya, wait. No break. And next 15 minutes the chapter will be over. Next 15 to 20 minutes the chapter will be over. Do you want me to take the break right now, or do you want me to finish the chapter in 20 minutes? Tell me. What is more important? 20 minutes, can you manage? Can you manage for 20 minutes? Yes. Because after 20 minutes chapter will be over. Then you take the break and come back for Gopika mam's class. Right? Now students, what do you need to add in your append of tube? This is called as your append of tube. Okay. PCR ingredients. What do you need to add? Okay. Yes. Now students, we have the PCR ingredients. We have the genomic DNA, that is your DNA to be added. Then we have your primers, which are your oligonucleotides, all of you know. Then we have forward primer and reverse primer. Then we have DNTPs. Now what is this DNTP, students? That is deoxynucleotide triphosphate. That is the building blocks. ATGC, simple. Then we have your Taq polymerase. This Taq polymerase is very important because students, this Taq polymerase is obtained from your Thermus aquaticus. Thermus aquaticus is a bacteria which is growing in hot springs. It can survive in hot temperatures. So you obtain this Taq polymerase from that Thermus aquaticus because this PCR cycle happens at very high temperature. Not all polymerase can survive in that high temperature. So we have the Taq polymerase. Now, so what do we have in the reaction? We have your genomic DNA, forward primer, reverse primer, Taq polymerase and DNTPs. And how do we get Taq polymerase? From hot springs, Thermus aquaticus, we get Taq polymerase. So we take a vial with reaction ingredients and we add it into our thermocycler. You're doing the experiment. You are doing the experiment. Remember, thermocycler. So what a thermocycler will do? Thermocycler will maintain the temperature and maintain the duration. Now we come to the steps. There are 3 steps. One is your denaturation, then is your annealing, and then is your extension. Repeat after me, students. Repeat after me, everyone. Denaturation, annealing, as well as your extension. Okay. Now what happens in denaturing, students? Denaturation is very simple. First of all, let me tell the temperature. Let me tell you the temperature. Pen is only not working. Great. Okay. Listen to me very carefully. Your entire denaturation happens at your 94 degrees Celsius to 96 degrees Celsius. 94 to 96 degrees Celsius. What happens in denaturation? In denaturation, your double stranded DNA becomes single stranded. Double stranded becomes single stranded because denaturation, very high temperature. In very high temperature, in 94 to 96 degrees Celsius, the entire double strand becomes single stranded in denaturation. Okay. Temperature is given here. Can you see? 96 degrees Celsius. Now next one is annealing. In annealing, what do? In annealing, we take our forward primer, we take a reverse primer, and we add it. Just done. Then we have extension. In extension, your RNA polymerase, your Taq polymerase will join here, and it will use DNTPs to extend it. That is your extension. And remember, students, your annealing happens at your 60 degrees Celsius. Extension happens at 70 degrees Celsius. Extension happens at 72 degrees Celsius. Now, once the extension happens, your DNA new strand has been synthesized. Clear? This is from your 3. Clear? And that is your entire PCR cycle. Now you will ask me, this PCR cycle will keep on continuing. It will keep on continuing until we get a desirable product. And there is a formula here. What is the formula here, students? Listen to me very carefully. To find the number of copies, what you need to do? 2 to the power n, number of copies. Now what is n here? N is the number of cycles. They can give you a small numerical here. Can I give you a small numerical here? Yes, I can give you a small numerical here. See the small numerical. After 4 cycles of PCR, how many DNA molecules are formed from one DNA? That is 2 to the power of n. What is n? N is the number of cycles. cycles. Yes. How many cycles do we have here? 4 cycles. 2 to the power of 4. Tell me in the chat quickly. What is 2 to the power of 4? Tell me quickly, what is 2 to the power of 4? That's going to be your 16. Done. Okay. Done. Easy question. Easy answer. Yes. PCR done. Yes, PCR also done. PCR is very easy. They'll not ask you many questions from there. Numerical can come. They can ask you temperature, or they can ask you about thermus aquaticus, or they can ask you the requirements of PCR. That's it. Nothing much. Okay. Then is your process of rDNA technology. That is insertion of recombinant DNA into the host cell. See, once you make multiple copies, we need to take our rDNA inside the host cell. That is, recipient cells become competent. That is your divalent cation, micro injection, gene gun, ballistic method, heat shock method. Done. Then we have your selection. Then you have selection. Then we have insertional inactivation to distinguish between your transform and non-transformant. Then we have done this, where we have ampicillin resistance, and we also have your blue and white screening. Yes. Remember this, rDNA is entering inside. Done. Now, once finally, students, after replication, all the replication, we have the transformed cell, which is able to produce your insulin protein. Example is insulin. Just taking an example. Finally, it is able to produce your insulin protein, and that is your insulin protein is the final product. Insulin protein is your final product. The next question is, students, why large scale production? See, all this, you tell me, bacteria will produce only so much. If you're doing this in the lab, if you're doing this experiment inside a lab, you will only get so much product. Will you get around 100 kgs of it? No. 100 liters of it? No. So you need to upscale it, right? You need to make more and more product. So that is why large scale production. That is not enough produce. TDF process which is require monitoring, and it is not cost effective. That is, if you do in a very small scale, it is not cost effective. You need to do a large scale. For that large scale, that is, how do we upscale the process? Can anyone in the chat tell me how do we upscale it? Now, you know, instead of doing it in the lab, now we are doing it inside a large vessel. How do you do it? Tell me quickly. Imagine you're cooking, your mother is cooking in the house. Your mother is cooking in the house, your father is cooking in the house, someone is cooking in the house. In your house, we have 4 people. Imagine in your house, you have 4 people. Your mom will make rice in a small cooker. Yes, your mom will make rice in a small cooker. Now imagine there are at least 50 people coming to your house. Imagine there are 50 people coming to your house. Will your mother make the rice in a small cooker? Tell me. Will your mother make the rice in a small cooker? No. She'll bring out a big container. She'll bring a big container. Right. Here also we have a big container now. That big container is called as your bioreactors. Before we go to bioreactors, you should understand, in large scale production, we have something called as upstream process. Also, we have something called as downstream process. Now, what is there in upstream process, students? In upstream process, we have your formation of seed stock, scaling up, and bioreactors. Now, what is the seed stock? Seed stock is nothing but what we learned throughout the chapter. Right? That is E. coli, making rDNA, making culture plate. That is comes under seed stock, culture broth. That is your seed stock. Then we have scaling up. In scaling up, we take the broth, we culture it in a fresh medium, and we take this medium, we make multiple copies of it, and then we add it into a bioreactor. So all of this is upscaling process, making your rDNA, restriction enzymes, getting your rDNA, transformation, vector, all of this is upscaling. And upscaling, the last part of your upscaling is bioreactors. Bioreactors is the last part of your upscaling. Here we have large vessel which is 100 to 1000 liters. And remember, students, what do we do in bioreactor? In bioreactor, we need some products. We need some raw material. What are the raw material here? Proteins, enzymes, and vitamins. Every single requirement for a bacteria to grow. For a bacteria to grow. That is, cell can be plant cell or animal cell. And this bioreactor will provide temperature, pH, substrate, salts, vitamins, oxygen. Yes. So what do we do now? We in your NCERT we have something called as stir tank bioreactor. In a stir tank bioreactor, there is a stirrer here. It will keep on rotating. When it keeps on rotating, entire mixture is being rotated. You have noticed, right? When your mother is cooking, she will keep on mixing it. Here also we are mixing it. We may keep mixing. Temperature control is there. Reactor tank, sample port where we add the sample inside. Then we have aeration. Why aeration? Because for the bacteria to grow, aerobic bacteria to grow, we need oxygen. Yes, oxygen is there. pH control, monitor. Then we have foam breaker also. Now, what is the use of foam breaker, students? When we are doing such experiments, there can be a formation of a layer here. There can be a formation of layer. To break this layer, we have the foam breaker. Now, based on that, we have single batch bioreactor, feed batch bioreactor. In single batch, once we add our reactants, once we add our enzymes, once we add our vitamins, once we add everything, it will be sealed completely and that's it. We have to wait. You cannot add any more reactant to it. Yeah? Your entire bacteria will grow from lag phase to exponential phase and to decline phase. But in the case of your batch, you can keep on adding the medium. That is, your bacteria will go from lag phase to exponential phase, but it will not go to the death phase. See, the main reason for death phase is because all the bacteria does not have nutrients. But right now, are we continuously supplying the nutrients? Yes, we are continuously supplying the nutrients. So your bacteria can keep on growing. Yes, it is single batch is safe, costly, time consuming. It is cost effective as well as takes less time. That is your entire upstream process. That is your entire upstream process. But what is your downstreaming process, students? What is this downstreaming? Downstreaming is very simple. See, once you take a tablet into the market, it has to go through many processes. That many processes called as your downstreaming. That is recovery and purification of product. Removal of undesirable elements formed by the cells present in the culture media. So basically, in your downstream process, students, in your downstream process, we are completely purifying it to make sure there are no mistakes. See, imagine example. I'll tell you one more example. In your insulin production, can we take any in the previous cases when insulin was taken from your cattle as well as pigs, the insulin gave some allergic reactions to humans because it did not undergo downstream process. If we had gone under downstream process, we could have removed all the impurities. Okay. This is basically the quality check. That is separation of cells, disintegration of cells, extraction and concentration, as well as your purification. Now, what do we do here? We find the right dosage here. That is, we find the exact dosage of how much is required. Then we also do the testing. Imagine the human testing is also the last stage of any particular product. And one more last one is going to be your cost effective. Imagine if I ask Gopika mam is bringing a product in the market. Ma'am, you come here. Okay. Gopika mam is bringing a product into the market. Gopika mam is selling the product for 10 rupees. You're selling a paracetamol for 10 rupees. Now I'll come. Oh ho, my product is amazing. My product is 1,000 times more effective than your product. But I'm selling my product for 1 lakh rupees. Same paracetamol I'm selling for 1 lakh rupees. Okay. Which one will they buy?
Which I'm selling.
Exactly why? It is cost effective. 10 rupees, 1 lakh rupees. They'll buy yours. Exactly. So when you're bringing a new product into the market, it should be cost effective also. It should be cost effective. Clear? Ma'am, say hi to everyone. I use as an example.
Oh, hi, how are you all? We have biotechnology application. Now they would have studied how to do it. Now they implement it in my class. Okay? Ready? It's a little slow.
Yeah. And the live watching is stuck.
Oh, is it? Okay. Hello. How much more time, sir?
5 minutes.
Then give them a lunch break.
I'll give them lunch break after 5 minutes. Or 3 minutes, ma'am, will be over then.
Then give them a lunch break. 20.
5 minutes.
How long you want the lunch break? 20, 25 minutes. Not more than that, huh? 20 or 25?
20 minutes, not 25 minutes. 20 minutes, I'll give them a break after this.
Yeah, sir. Okay. Continue, sir.
Okay, thank you so much, ma'am. So again, in quality check, what do we do? Steps to maintain the identity and purity. Helps to maintain the quality check and vitality. There are 3 steps. Determining the composition, checking for adulteration, and checking for packaging. And students, with that, your entire chapter is over. With that, your entire chapter is actually over. Do you realize in 2 hours, right? 2.5 hours. In 2.5 hours, we have finished the entire chapter. We started 11:30, 12:30, 1:30. Yes. 2.5 hours, the entire chapter is over. And we have cleared the entire concepts of your endonucleases. What is restriction enzymes? What is PCR? What is selectable marker? Everything is done and dusted. Yes. Do you see how beautiful this chapter was? Do you realize how beautiful this chapter was? Everything is done. And students, if you have any doubts, sir, recombinant technology is very easy. Sushmita, you can just watch the video in 1.5x speed, you will understand the entire thing. Just watch it in 1.5x, you will understand everything, you will understand. This I've gone very, very slow. I've gone very slow, but with killer visualization. Okay. Recombinant DNA, you understood, right? Recombinant DNA, what do? The first slide, remember recombinant DNA, I told you. In recombinant DNA, we take a particular gene of interest, cut the DNA, isolation of DNA. Once we isolate the DNA, we add it to the vector. Now, once we add to the vector with the help of restriction enzymes, then we have recognition site, which is also called as your cloning site. Once you have the vector, add to your host. Once we add to the host, what do? That is called as transformation. There are multiple methods for transformation. That is your heat shock method, gun method, ballistic method, right? And micro injection. Done. That is your entire process. That is the entire process. Now the question is, do you want to solve some questions? I have some 3-4 questions for you. Do you want to solve some questions? Neet PYQs. Some Neet PYQs. And students, trust me, after today's class, every single point from this chapter will be clear to you. You will be able to solve every single question. I think I missed out one point. It is sparge tank bioreactor. In your sparge tank bioreactor, from the bottom itself, there is release of oxygen. In sparge tank bioreactor, bottom itself, there is release of oxygen. Okay. The next is going to be your cell unit. Yes. The next question is going to be your cell unit. Block 4 cell. Block 4, we have the cell. Okay. Question here. This is a Neet PYQ, Neet 2023 PYQ. Upon exposure to UV radiation, DNA strained with ethidium bromide will show. Alright, orange color. During purification process of recombinant DNA technology, addition of chilled ethanol precipitates out what? RNA, histone, polysaccharide, DNA. DNA answer. Next question. In gene gun method used to introduce the alien DNA into the host cell, microparticles of dash metals are used. We just learned we use your tungsten and gold. Yes, we use tungsten and gold. Done. This question is very important. This question is very important, friends. This question is very important. Main steps in formation of recombinant DNA technology given below. Arrange the steps in your correct sequence. Arrange the steps in the correct sequence. Now here I'll tell you where students have gone wrong last year. They have gone wrong between these two options. Now try to solve this and I'll tell you where you can go correct. Sir, in transformants divide into recombinant and non-recombinant. See, Jay Ganesh, understand, we can divide transformant and non-transformant. Yes, transformants will have your vector. Non-transformants will not have vector. Non-transformants will not have any vector. Okay. Under transformants, we have recombinant and non-recombinant. Under transformants, we have recombinant and non-recombinant. Okay, recombinant and non-recombinant. Recombinant will have vector plus gene of interest. Non-recombinant will not have gene of interest. That's all. Okay, what is the answer? Answer is 2. Answer is 2, not 4. Answer is 2, not 4. Let me tell you why. Let me tell you why. Now, if you look here, it says isolation of desired DNA fragment. It says isolation of desired DNA fragment. Now, how do you isolate DNA fragment? Tell me in the chat. How do you isolate DNA fragment? See, first step is what? First step is isolation of gene of interest. First step is isolation of gene of interest, right? Isolation of interest. We obtain gene of interest by doing restriction endonucleases. Then you get a gene of interest. But here it says isolation of desired DNA fragment. Isolation of DNA fragment. How do you obtain DNA fragment? DNA fragment isolation is done with the help of gel electrophoresis. Gel electrophoresis. Yes, not restriction endonucleases. So first is what? Cutting the DNA at a specific location by restriction enzymes. After cutting it, we get the fragment. After that, we get the fragment. That fragment can be isolated by gel electrophoresis. Then we have your amplification. Then we have insertion of DNA into the host. Okay. Okay. Next question. This is homework question. This is your homework question, all of you. This is your homework question. Don't tell me the answer. I want to see the answer in the comments. I want to see the answer in the comments. Oh my god, not even 3 students. Quickly like the video if you have watched, if you have understood the concept, if you're able to solve the questions. Quickly like the video, everyone, because I wanted 300 likes as the target, but let's see. And I know KCT is tomorrow. And I know KCT is tomorrow. Okay, I know. And someone was asking me where I've studied from. I have studied from KVISC. All of you know, some of you might know. This is my school. This is my school. Yeah, this is my school. Someone was asking, right? I saw someone asking. Telegram, you can just write Basavaraj sir biology. You will get my Telegram, where I'll be adding more questions today. And if you want to solve more questions, join the Telegram. Okay. If you want to solve more questions, join the Telegram today only. Okay. Basavaraj sir biology. You know my spelling, right? Okay. All of you know IISc, right? Indian Institute of Science. Indian Institute of Science. IISc, KV IISc. Now we'll take a break. Class will resume at 2:35. Okay. Okay. There is a question. There is a question. There is your answer. And yeah, Mentiquiz will keep all the best to all of you. All the best to all the KC students. Chanag Madi. Exam ready. 1 second. This is your break time. You can go. Bye bye. So before I go, can I tell you something? Before I go, can I tell you one small thing? Students, how many days do you have for your examination now? Tell me. How many days do you have for examination now? Tell me. How many days do you have? For your examination, how many days do you have? For examination, how many days do you have? Tell me carefully. We have so many days left. Do you have a lot of days? No. We have very few days left for your examination. And the best thing right now would be to solve many questions. Right? The best thing right now would be to solve many questions as much as possible. Right? Many questions as much as possible. So how do you solve such questions? Do you have to buy a book? Sometimes you can buy a book. Yes. But is there any easier method where you can solve many questions? Easier method? Yes, there is an easier method. That is your All India. What is happening? That is your All India mock test. All India mock test series. With the help of this All India mock test series, you can actually solve free papers. Free papers. So there is, do you see this particular link here? If you go on the link right here, there's something called as AIPMT series, All India mock test series. If you click here in the All India mock test series, we get a page like this. We get a page like this, All India mock test series. Now, what you need to do? Do you have to pay for this? No. This All India mock test series is completely, absolutely free for you. Absolutely free of cost. So if you just click on your enroll for free, you'll obtain a page like this. And if you actually enroll here, right, if you enroll here, you will be able to write free mock test on your own time. And this mock test is completely online. You can take the test today also. And remember students, in at least the last 15 days, if you do not solve every single day 2 mock tests, you will not get the feel of this examination. If you want to get the feel of this examination, for at least the last 15 days, solve as many questions as possible. Solve as many questions as possible in the last 15 days. So students, have you enrolled for this? Tell me in the chat. Have you enrolled for this? Tell me in the chat. Have you enrolled? Quickly tell me in the chat. Have you enrolled for this free mock test? Because if you have not, you are missing out on something. If you have not, you are missing out on a majority of things. So please don't miss out. Don't miss out on such free opportunities. Take advantage of that and you will get your dream medical college. Okay. Let me write here one more time. Class will resume when? Okay. So students, enroll today. Take advantage of, see how much ever we can, we are giving you. We are teaching you for free here. We are doing for everything for free. So take advantage of everything. Okay. One second, students. Sure, then I'll wait Gopika mam to come now.
Fine. Okay. Awesome. 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 So students, I told you, do not ignore this chapter. This chapter is very, very easy, and this chapter has a potential of giving 6 questions, because in the past, in 2020, your COVID year, there were 6 questions from this chapter. So we can expect that 4 to 5 questions can come this year also, and easily in 2 hours, 2.5 hours, you can finish this chapter. So in 2.5 hours, if you're getting 24 marks or 20 marks or 16 marks, what more than that a NEET student want? Yes or no? Okay, more than that, what do a NEET student want? So let's start the class, students, no more delay. So I was telling that biotechnology is basically any problem humans face, right, to make our lives more easier, right? To make our lives more easier, we need this biotechnology. And you also studied in your principles how we can modify to make genetically modified crops or genetically modified organisms, or you want to do transgenic animals. For all this, we need this biotechnology applications. Not only that, when a plant, okay, biotechnology and applications is basically the chapter is divided into 3 sectors, right? So in that also, how we can improve the agriculture, okay, how we can improve the medicine. These are all discussed in this chapter, right? These are all discussed in this chapter. So we are expecting 4 to 5 questions from this chapter, and 2 questions I'll tell you today. 2 questions, 8-mark question confirm, I'll tell you today. Okay, so stay tuned. Okay, for those who liked the starting of the class, those who joined right now, quickly smash the like button. Quickly smash the like button, all of you, and mark your attendance. Now, students, very, very important thing. Those who are missing this lecture will not know these 2 things. What is that? From strategies chapter, okay, from strategies chapter, 2 things have been added from there to here. All of you know that strategies is deleted. So most of you, is there a problem? Okay. So students, strategies chapter, 2 things have been added here. First thing is your tissue culture. Okay, first thing is your tissue culture. So most of the students who have the old NCERT might skip this. Okay, most of the students who have, okay, the old NCERT will not see this tissue culture in your textbook. Same way, you will also not see somatic hybridization. Students, don't worry, we are doing NCERT line by line only. Okay, we are doing NCERT line by line with my notes. Okay, yes. So these 2 topics are in right now in your new NCERT, but it is not there in your old NCERT. So where will you learn it from? Go to Google, type biotechnology and its application, get the new PDF and study from there. Is this clear? Is this clear? Do not, okay, did I write tissue paper? Oh God, tissue culture, yeah. Okay, so did I tell, what are these people talking? Okay, so do not skip this, okay, tissue culture and somatic hybridization. Got it? Yes? So look here, these 2 topics not there in old NCERT but present in new NCERT, and this 2 has come from strategies chapter. Okay, strategies chapter. So today we'll be doing this because these 2 topics have been taken from there and put here. We can expect each of them 1 question, yes or no? So we will make sure that we study these 2 topics thoroughly. Okay, students, let's start with the chapter. This topic starts with green revolution. So I'll tell you, this chapter is divided into 3 parts. First we start with agriculture, then we go to medicine, then we go to transgenic animals. Okay, all of you got it or not? Yes, definitely. You can read it from strategies or from Google. Go to Google and get the new PDF and read it from there, because I don't want anyone to lose marks because you are not aware of it, right? So look here, first thing about biotechnology, like I already mentioned, here let's see what NCERT is telling. See, providing the best catalyst, okay, providing the best catalyst in the form of improved organism, usually a microbe or a pure enzyme. So where are we using all the thing that you studied in your principles? You're using it in application chapter. You're going to implement it in day-to-day life to make human life better. Okay, to make human life better, to satisfy human greed, need, or even requirement. Sometimes even it's a blessing because it can cure so many diseases. So look here, providing the best catalyst in the form of improved organism, usually a microbe or a pure enzyme. Now, creating optimal condition through the engineering for a catalyst to act, downstream processing technologies to purify protein and organic compound. All this clearly you have understood in your principles chapter. Yes or no? Yes or no? Okay? Yes. Fine. Now we are going to get into the actual chapter. So the actual chapter starts with first called as something called as Green Revolution, which was worldwide. The father of Green Revolution worldwide is Norman. Okay. Norman. And in India, okay, in India, the father of Green Revolution, can anyone tell me in the chat? Swami something? Can you give it? Yes, very good, Norman. Yes, Norman Borlaug is worldwide known as the father of Green Revolution. Can anyone tell me? I cannot see this far, so I'll see it here. Yes, tell me. Yes, very good, very good. M.S. Swaminathan, right? M.S. Swaminathan. So students, these two people are known as the father of Green Revolution. Now, what happened during Green Revolution? Very simple thing. Let's see here. The Green Revolution succeeded in tripling 3x times, okay? 3x times it improved, okay? Tripling the food supply, but yet it was not enough to feed the growing population. So students, what is happening? India growth, right? You know, population is exponentially growing, and even though this Green Revolution focused on, what did Green Revolution mainly focus on, students? High yielding, okay, 1 second, I'll move after writing. High yielding crops plus the best chemicals will obviously give us good results. Yes or no? Now for you good mentors, right, for you, if you get good mentors and good mock tests, okay, if you get good mentors and good mock tests, will you be happy or not? Will you be happy or not? You'll be very, very happy. Yes or no? Same way, this Green Revolution focused on doing what? Green Revolution focused on using high yielding crops with chemicals so that there will be no pesticide, there will be no weedicide, right? None of this will be there, and this will also improve what? This will also improve what? Yes, this will also improve the amount of crop that is going to happen. But even though this improved to 3x, it was not helping the population. It was not helping the population. Okay, so look here, it was not enough to feed the growing human population. Increased yield have partly been due to the use of improved crop varieties. That's what I told you, high yielding crops. Yes, okay, but mainly due to the use of better management practices and use of agrochemicals like fertilizers and pesticides. Okay, however, for the farmers in development world, agrochemicals are often too expensive and further increases in yield with existing varieties. So Green Revolution focused by Norman and your Swaminathan. And student, there's also a political father for your Green Revolution. If you read a lot about it, it's Chidambaram. Okay, I love the name Chidambaram. It's such a beautiful name, so gorgeous Indian culture name. So when I read it, I was like very happy. So the political father of Green Revolution is Chidambaram. Okay, in case you come across and you're like, ma'am didn't tell about Chidambaram, it's a political father, okay, of Green Revolution. So quick recap, Green Revolution could actually increase up to 3x, but it was still not enough according to the Indian population. Okay, it was not, I mean the entire world population, the growing population was so fast that it could not feed or it could not be sufficient. And what did they use? Simple thing about Green Revolution, they basically took high yielding crop, used better chemicals or fertilizers or pesticides to, you know, protect the plant. So good results were possible. Are you all okay? Yes? Yes? Clear? Yes. Chidambaram Subramaniam. Fine. Now students, first we learn from NCERT, then I'll write by myself. Okay. Now tissue culture. This is the topic which is taken from strategy. So look here. As a traditional breeding technique failed to keep the pace with demand. All that is not required for us. See, what is tissue culture? Students, simple thing. There is a test tube. Okay, there is a test tube. Can you all see if I draw here? Yeah. See, there is a test tube like this. In the test tube, I will put an explant or any small portion of a plant. Okay, it could be a cell, it could be a tissue, it could be a meristem, it could be any of this. Okay, an explant is put and I will provide this explant with all the nutrients that is required. Okay, all the nutrients. See, now I am giving you so much free content, still you people complain. Ma'am, did you say 5' to 3' or did you say 3' to 5'? Is that a big doubt? Is that a big doubt? Why can't you just check? Why can't you read NCERT? Right? So, all this. Okay, so students, look here. There is test tube and there will be an explant put into that. For that explant, I will provide everything that is needed. That means I'm going to provide the micronutrients, macronutrients, everything I will provide. Okay, everything I'll provide in this tissue culture. And what is going to happen? The plant is going to grow. Right? The plant is going to grow. So, those who spam the chats will be out of the class very soon. Okay, our team is sitting behind this, so they will remove you immediately. Got it? Okay, see, I told you. So sharp my team is. See how fast the message came? Okay. Yes, look here, as the traditional breeding technique failed to keep the pace with the demand and provide sufficiently fast and efficient system for crop improvement, another technology called as tissue culture got development. Now, what does tissue culture mean? What does that mean? I just told you right now. It was learned by scientists during 1950s. Students, years are important. Okay, 1950s, that the whole plant, okay, whole plant could, one second, the whole plant could be degenerated from an explant. Now, what is an explant? It could be a small portion of a cell, a tissue, anything that has the ability to develop into a completely new individual. Right? This is not new for you. This tissue culture you have studied in your previous classes. Okay? That is any part of the plant taken out and grown in a test tube under sterile condition. So, how many of you have seen a tissue culture lab, students? How many of you have seen a tissue culture lab? Have any of you visited from school or in your PU days? Have any of you visited? Yes? Yes? Yes. You will get all this PDF on Vedantu Neet English. So, quickly go to Telegram, search Vedantu Neet English, around 10K plus students we have. All the PDF will be sent there. Okay? Yes. Yes, ma'am. Awesome. Awesome. So, students, you would have seen a tissue culture lab. How would it look? So, there will be many test tubes like this kept. Okay? Many test tubes will be kept. All of them will be kept and inside that explants will be there and there will be lighting systems will be there and every single person who enter the tissue culture lab will be wearing gloves, lab coat. Why? Because the entire environment is sterile. Right? Entire environment is sterile. So, whatever is there in the soil, better quality content will be given there. Okay? Better quality content will be given. Be it micronutrients, be it macronutrients, be it whatever is there in the soil, soil layers, everything will be provided for this plant. Okay? Now, under sterile condition in special nutrient medium, very, very important. This capacity to generate a whole plant for any cell or an explant is called as totipotency. That means totipotency means a plant's ability or a cell or an explant's ability to grow into a completely new individual or differentiate into a completely new individual. Any doubts so far? Okay. Sterile means what? That means completely neat, clean, not like just cleanliness. That means they are completely, wiped with alcohol, ethanol, so there are no germs. Entire place is, sterilized with alcohol, so there is no, fungus, bacteria or any other infection that can happen because it's laboratory, it's not, a farm. So, we have to take care like that and every single person will wipe their hands with alcohol, with rubbing alcohol and enter. No nails are allowed. So, like that so many situations are there for the sterile condition. Okay? Yes. Yes, clear? Okay. Okay, so totipotency means a cell's ability or an explant's ability. Why am I writing like this? I'll write like this. Explant's ability to differentiate into a completely new individual, right? Differentiate into a completely new plant. New individual. Is this clear? This word is important, students, because this is taken from strategies. Very high chance that one question can come from here. So, those who are talking with each other, You will never study this. Okay, so focus here. Now, totipotency understood. Now, you will learn how to accomplish this in higher classes. Already you are old enough. Let's not talk about this. Now, very, very important thing. What are the materials that I've given in this nutrient medium? First thing I will give is what? I am going to give your carbon source. That is my first ever content that I'm going to provide. That means food for the plant, right? For this ex-plant to grow, I need to give the nourishment. So, first thing I'm going to give a carbon source. Second thing, such as sucrose and also. Second thing, inorganic salts. Third, vitamins. 4, amino acids. 5, growth regulators like auxin and cytosine. So, they give PGR, inorganic salts, they give vitamins, they give amino acids, they also give carbon source, out of which carbon source is the most important one. Right, food we need to give. Okay, so these 5 things, students, they can ask you which are the, what are the medium, what is there in the medium. So, you should be in a position to answer all this. Fine. Now, like auxins and cytosine, by application of this method, it is possible to achieve propagation of a large number. So here, what are they talking about? They're talking the benefits of tissue culture. Okay, they're talking the benefits of tissue culture. First thing, what they can do in one laboratory, I can do multiple plants, right? I can grow in large numbers. Yes or no? If you are a farmer, all of you think like a farmer. All of you think like a farmer. You are a farmer. Okay. Imagine you had one kind of a cultivation. Now, because of some kind of, now imagine Bangalore has water crisis, okay, or somewhere in the outskirts there is water crisis. Suddenly you will not be in a position to change your complete crop because you have to wait till the harvest. So you will be completely in a dilemma. You will be like, no, okay, what do I do? What would you do if you are a farmer? You have, grown a crop. You thought that this is going to give you a good harvest, but so unfortunately, some of the abiotic or biotic factors have changed against it and you are sad. Yes or no? Yes? Okay, you will be feeling very disappointed. You cannot shift suddenly. But in tissue culture, I can do that. Why? Because I am using a tissue, I'm using a small test tube. In this test tube, I'm putting some nutrient medium and I'm cultivating. So in large scale, I'm growing. Right? In large scale, I am growing. So look here, the first thing is large numbers of plants in a very short duration can be grown. Can this be done by a farmer? Can this be done by a farmer? Not possible, right? Now, this method of producing thousands of plants through tissue culture is called as micropropagation. Okay, so important. Doing this method, okay, by growing this 1,000 plants in the name or in the process of tissue culture is called as micropropagation. That means using a small portion, you propagate or you differentiate to grow a completely new individual. Now, each of these plants will be genetically identical to the original plant. The second benefit, don't worry, I'll write all this. Okay, second benefit, genetically identical, or your soma clones are got. You have got soma clones from this, right? Now look here, original plant from which they are grown. That is, they are soma clones. Any doubt? Any doubt? Okay, good job. Fine. Many important food plants like tomato, banana, apple, all of this have been grown, okay, this way, have been produced on commercial scale using this method. Okay, now try to visit, okay, that you have to listen to NCRT. Now, second most important thing about tissue culture, students. Okay, ma'am, you told that, okay, I can do it on large scales, I can form soma clones. Ma'am, what about deceased plant? You would have thought this, no, if you're a farmer, right? If you're a farmer, you would have thought, ma'am, what about deceased plants? What am I going to do about that? Now, you all know this red rot in sugarcane, tubers in your potato. So many problems are there. So many kinds of diseases are there. So many kinds of infections are there for the plants also. Now, to deal with this, can tissue culture help? That's your question, right? Look here. Whenever a plant has a disease, okay, any kind of infection, if it has not affected the meristem, it will not affect the meristem most of the time. If it has not affected the meristem, I will use the meristem as an explant and I will grow the entire new plant. Problem sorted? So here the statement says, see, method is recovery of healthy plant from a deceased plant. Recovery of a healthy plant from a deceased plant. How is it? Even if the plant is infected with a virus, the meristem or the apical, anything, apical or axillary meristem is free from virus. So I will take that as an explant and I will cultivate. Okay, so hence one can remove the meristem and grow it in vitro to obtain a virus-free plant. Is this clear to all of you? So the question can come, what are the uses, or which among this is the correct statement, and they'll give you all the uses of tissue culture, or they can give you all the incorrect statements and find one correct statement. So you should be in a position to tell all these points. Clear? Right? Okay, now scientists have succeeded in culturing meristems of banana, sugarcane and potato. So students, remember banana was there in both the places. See, look here, tomato, banana, apple, etc. So here what was there? TBA was there. Okay, you can think ABT also. ABT was for normal tissue culture. That means take an explant, because of its totipotency it forms a completely new plant. Here what is happening? Here you have SBP. Yeah, SBP. This is meristem. That means diseased plant we have taken and we have grown into a healthy plant. Okay. Yes or no? Okay, fine. Got it. Now, let's write what we studied now. Ready? All of you ready? All of you ready or not? Where did I go? Yes, so far good students, understood? Any doubts? Any doubts? No, right? Yes, shall we write what we just studied now? Chair is giving reflection, no? One second, students. Okay, done. Chair is out of the frame. Okay, otherwise tomorrow you people will complain. Ma'am didn't even see the chair. Reflection came. Yes or no? Okay, let's write. So students, look here. What did we do? Tissue culture. First we understood terms like explants. So I took any high yielding. Okay, I took any high yielding, or I took a deceased plant. Deceased. And I used what? I took one portion of it, and that portion is called an explant. Right? I took an explant. And because of this, nature of this explant to show totipotency. Okay, totipotency. I was able to form a completely new individual. Okay, completely new plant. Yes or no? Completely new plant. This is basis of your tissue culture. Now, what exactly did I take as an explant? Right, what did I exactly take as an explant? I took, see, I'm writing it here. I could take a cell, I could take a tissue, or I could even take a meristem. I could even take a meristem. This is what I've taken as an explant. Right, now all of this I kept it in a medium. Okay, all of this I kept it in a medium, and that medium let me grow this. Now, what did that medium contain? All of you tell me, what was the things that I added into the medium? One by one, all will tell me what were the things I added into the medium. Okay? Yes. Quickly, fast, fast, fast. Answer should come faster. Okay, this much you cannot wait. You cannot expect me to wait. Yes, very good. By then I'll draw a tissue culture setup. Yes, will you all tell one answer? Yes, very good. I cannot see you only this far. Okay, yes, very good, awesome. First main thing is carbon source. I could have put anything, a sucrose, anything that is mentioned in the NCERT. Next thing what I could add inorganic salts. Okay, inorganic salts. Then you people told amino acids. Okay, amino acids. Then what else, students? Did any of you tell vitamins? Did any of you tell vitamins? Yes, very good. Cypria has told vitamins. Many of you have told mega. So vitamins. And the last most important thing is your PGR, that is your plant growth regulator, and two things is mentioned, auxin and cytosine. Auxin and cytokinin. Cytokinin. Okay. Yes. This is what we added, and this is all that makes the nutrient medium. Okay. This is what the entire. This is the entire what is there in my nutrient medium. Okay, nutrient medium. Now, in this side, let's write what are the advantages. What are the advantages of tissue culture? All of you, one by one, all of you tell me. One by one, tell me the advantages of tissue culture. Here I am writing. So, this page will be more than enough for you to study the whole thing. Okay. Advantage. First thing I am going to write. Okay. One thing you will tell. I am going to write the first thing what? It is to grow in large scale, right? To grow in large scale in a shorter period of time. Okay. Large scale production. Large scale production. Now, next point, all of you tell me. Large scale production in shorter period of time. Very good, very good. Genetically identical plants. What is the other name for it? Genetically identical plants. The other word is somaclones. Okay. And the last one more point which is coming as a benefit. Yes, disease resistant is there, pest resistant is there, all that you can write. But exactly what did we study now? We can also make a diseased plant healthy plant. Yes or no? Yes, correct. Okay. Diseased plant, from a diseased plant, we can make a healthy plant, right? From a diseased plant, we can make a completely healthy plant. Okay. This is by taking the meristem. Is this clear? This entire page is more than enough for you to study your entire tissue culture. I'll also, okay, this is your entire tissue culture. Any doubts so far? Yes, recovery from diseased plant to healthy plant. Student, other than this, there are a lot other benefits. Obviously, it will be, pest resistant, genetically modified, though it will be obviously pest resistant. It will also be high yielding plants. Okay, but you have to tell exact NCRT line, then this is your NCRT line. Okay, yes, fine, good to go. Sorry, yes. So now next topic that has been added again is somatic hybridization. Let's understand what is this. Very similar but a little different. Okay, see, look here. Scientists have been isolated single cells from plants after digesting their cell wall. Very focus here. Single cell, they have been taking two varieties and they have been digesting their cell wall. Okay, digesting their cell walls. Have been able to isolate naked protoplast. Okay, what is protoplasm? Protoplasm is basically your nucleus plus cytoplasm. Any doubts in this? Nucleus plus cytoplasm, right? Here what are they getting? Isolated naked protoplasm. That means without the cell wall. Now, surrounded by the plasma membrane. Isolated protoplast from two different varieties of the plant, each having a desirable characters. That means I want to form this kind of a rose. I want to form this kind of a hibiscus, or I want to form this kind of a, I want to form potato plus tomato. Now for all this, I will have to do somatic hybridization because I love potato. Okay, I love French fries, I love potato, I like eating anything with aloo. I also like anything with tomato. Now I'm like, okay, who is going to, cut them separately, eat them separately, so I'll just mix it. So I will make a pomato. Now if I want to make a pomato, what am I going to do? I'm going to take the variants, I'm going to digest the cell wall, I'm going to mix that, form a hybrid. Okay, so look here, each having a desirable characters can be fused to get a hybrid protoplast, which can be further grown to form a new plant. These hybrids are called as somatic hybrids. Okay, the one that we formed is called as somatic hybrid, while the process is called somatic hybridization. Imagine a situation when the protoplast of the tomato, look here, is fused with that of the potato. This gave a pomato. Actually, lot of people love, especially in the north. I think in the south is comparatively less people, mixing potato and tomato curries. But now it's become very common because, COVID time we all investigated and found a lot of dishes. So people love that kind of a curry. But imagine eating tomato, and if it is not successful, then the taste is not met, then people will not buy it, right? People will not buy it, and then they are grown to form a hybrid plant combining tomato and potato characteristic. Well, this have been achieved, resulting in the formation of tomato. So it's not like it did not happen. Tomato happened, but it lacks something. Okay, unfortunately this plant did not have all the desired combination of the characteristics. Got it? It was not commercially used. It was not like I could not sell it. I could make tomato, but I could not sell it because it did not have everything that I required. Got it? Fine. Let's write now what we learned. All of you, quickly. Tell me, what and all did we learn? One second, students. Yes. Tell me, what is somatic hybridization? Why is the number so disappointing? Okay, all of them asked for this chapter and boom, vanished. Yes. Okay, tell me what is somatic hybridization. I'll just get the water bottle. Yes. So students, I forgot. There is something super duper exciting for all my Tamil pasangal, okay? All my Tamil beautiful people out there. There's something super exciting coming today on this channel, mostly at 6 or 7. So stay tuned. Do not miss that session. I'll be coming and revealing some big surprise. I think most of my students will be excited. So how many of you are from Tamil Nadu? Raise your hands in the chat. Raise your hands in the chat. How many of you are from Tamil Nadu and are you excited? Okay, today 7, something big is going to come. Okay, for other students, slowly it will come. But right now, it's going to first to the Tamil Nadu students. Let me see. Let me see how many of them are excited. Slowly, we'll come to other places. As of now, Tamil students will be very, very excited. Okay. Do not miss the live and share with all your friends. Okay. Chalo. Okay. So, students, somatic hybridization, very simple thing they did. They took 2 variants. Okay. We'll keep it as variant 1 and they took variant 2. Okay. Variant 2. And what did they do? They digested the cell wall, right? Cell wall was digested. Okay, let's write it like this, more easier. First, we will take the protoplast, extract the protoplast, then we will write this, okay? Excited? Excited? What a guessing they're doing. Next, amazing guessing. So smart they have become. Okay, look here students. 2 variants. We took protoplast of each of them. Okay, protoplast 1. I want all of you to write this with me because if you write this, you will not have to read a big paragraph. Protoplast 2. Okay, how did I get this protoplast? Obviously by digesting the cell, right? Cell wall was digested. Okay, digested. Now, look here. This 2 protoplast, I'm going to fuse now. Okay, this 2 protoplast, I'm going to fuse and I'm going to get a hybrid protoplast. What am I going to get? A hybrid protoplast. This hybrid protoplast will finally form my new plant. Okay, and this new plant will have all the desired characters I want. Okay, all the desired characters. Yes. Good to go. Good to go. They're giving farewell for one student. He's telling bye and going. All are like, oh, bye. I'll miss you. No, concentration span is very less. Yes, digesting the cell wall. Okay, now I'll ask you a question. The question is, tell me the example of the plant that we can, okay, the plants that we have taken meristem, okay, and saved them from diseases, or from diseased plants we have made them healthy plants. Put the examples quickly in the chat box, all of you. So somatic hybridization, clear. Variant 1, variant 2, protoplast 1, protoplast 2. Okay, we'll get a hybrid. Okay, we can also call this hybrids as somatic hybrids. And the process is called as somatic hybridization. Chalo. Yes, how many of you got it right? Banana, sugarcane, potato. Yes, very good. Okay, how will you think? SBP you can remember, or BS, BPS you can remember. Okay, yes, SBP, BPS. BP is easy, blood pressure, you will have it in the exam. Okay, blood pressure you will have it in the exam. So BP you can remember, and S you add on. Okay, with your name you add off. Yes, very good. Okay, good, awesome. Now students, let's understand genetically modified organism or genetically modified crops. What are these people? Okay, what are they going to do actually? Look here, plants, bacteria, fungi, animals whose genes have been altered. So genes have been altered by manipulation are called as genetically modified organism. Is this clear? Any organisms, if their genes have got altered or modified by manipulation, those are called as genetically modified organisms. So GM plants have been useful in many ways. Genetical modification, now let's see how they have actually benefited over time. First thing, made crops more tolerant to abiotic stress. Now students, this is one main problem that every single farmer is facing. Yes or no? Okay, because if any of this happen, cold or salt condition has increased, any of this happen, the farmer is in a very bad position that he cannot just change the crop over day. But this genetically modified crops have been resistant toward that, because of which they are able to form high yielding varieties. Okay, now reduced reliance on chemical pesticide, very important, because even before genetically modified organisms coming, people have, plants also, people have actually tried to increase the pesticide, increase the fertilizer, increase the weedicide, spraying in the soil to get, better crop rotation to happen or better crops to be yielded. But what has happened over time? Humans are affected by human activities, or anthropogenic activities have affected us over time, that we have got such, so many genetic disorders, so many, skin issues, so many other multiple problems, right? So sometimes we are our own enemies. Now that can be reduced, right? Chemical pesticide help to reduce post harvest loss. Most of the time, if you have ever watched a talk show with a farmer, they will always say that after harvest my heart breaks seeing the post harvest loss. Yes or no? That also can be reduced. Now, increased efficiency of mineral usage by the plants. This prevents early exhaustion of the soil fertility. Have you all, in geography we have studied, no? What is that condition called? Slash and burn, right? All of you remember? All of you remember? In geography you would have studied slash and burn. That means the entire place where the crop was there, they will burn that entire place, leave it for 1 year period of time, so the soil fertility increases. Now the farmer is, if a farmer has a very less acres of land or very less land, that farmer's entire family will go into a very bad position financially, because that entire place is let for 1 year to increase the fertility of the soil. Can we afford that every time? Not possible, right? So that way this is a good thing. Now, enhanced nutritional value of food like golden rice, vitamin A enriched rice. Now we'll learn that only. Okay, now we'll learn that. See students, golden rice. I don't know how many of you have heard about golden rice. Yes or no? Yes. Yes, Cherry. We completed biotechnology principles and processes. That's how I started the class. Okay, students, look here. Gene addition. Gene addition means I'm adding a gene to modify something among it. And the first thing I'm going to do is golden rice. That means I want to improve the nutritional enhancement. I want to improve the vitamin A. Okay, that is what is happening. Now, all of you, this diagram might not be that clear, but you can see this is a daffodil plant. From the daffodil, we are taking beta carotene. The gene taken is beta carotene. And this is what you have studied in your principles. Now, you are going to use it here. Okay. See, look here. The genes are taken, it is inserted into your plasmid. Plasmid of which bacteria? Agrobacterium tumefacium. Okay, that agrobacterium will be allowed to, infect the embryo, that means the rice, and then from there the growth will continue. Now, what has happened here? Okay, what has happened here? Students, golden rice, or how it happened was simple rice plus beta carotene. Okay, beta carotene. This beta carotene is actually taken or extracted from daffodils. All of you know daffodils? And plus Erwinia bacteria. Okay, Erwinia bacteria. Now, this normal rice with this beta carotene, okay, that is extracted from your daffodils, and Erwinia bacteria also is helping. Okay, we are going to get golden rice. What are we going to get? Golden rice. Okay. Now, I just realized one thing, that I don't know what is that one thing. 1 second, students. I'm just thinking what is that one thing. No. Okay. I was just thinking if your NCRT order is the same. Okay, students, look here. Rice plus beta carotene, which is extracted from your daffodils and Erwinia. Now, what is happening? This rice plus beta carotene is going to give me golden rice. It's going to give me golden rice. Now, why did I do this? What was the need for me to form the golden rice? The need is very simple. I wanted a rice which is vitamin A rich. Okay, vitamin A rich. And this was only possible if I added a gene. Okay, if I added a gene. Okay, yes or no? Yes, RNA interference we will come. Don't worry, RNA interference we will come. Don't even worry. But is this clear to all of you? Is this clear to all of you? So students, very simple. Rice, normal rice. To that I have taken what I have added, which gene I have added? Beta carotene, which I have extracted from the daffodil plants. That I have added to form the golden rice. Now, why did I form the golden rice? Because I wanted a vitamin rich or a nutritional enhancement was required. Now, how did I do this? I took the help of, obviously, your agrobacterium. Okay, obviously your agrobacterium. Now, this you already understood in your principles. Yes or no? Yes or no? Yes, fine. Any doubts? So here I told you the genes into the plasmid, that plasmid into agrobacterium, again to the embryo, and the golden rice is formed. So student, this was, why did we do this? Mainly because this vitamin A, no, deficiency of vitamin A, deficiency can cause, okay, deficiency can cause many diseases. Main one, color blindness. So people with color blindness, I could just give them golden rice and cure it. And that is the best thing about this genetical modification or gene addition. Not only that, people who have cirrhosis, okay, xerothalmia, like that, many issues could be cured just by increasing the vitamin A content. And what did we do? Very simple thing we did. We just added a gene with the help of a bacteria called Agrobacterium tumefacium into normal rice, and we got golden rice, right? And this golden rice was rich in vitamin A, which could which could cure all the deficiency diseases. Is this clear or not? Is this clear or not? Yes or no, students? Tell me quickly. Yes, color blindness, night blindness, no, sorry. You can color blindness, night blindness, okay? Night blindness. Done? Okay, this is nutritional enhancement. Okay, this is one technique where we are using nutritional enhancement. Okay? Where do I write? Where do I write? This is one technique we use for increasing the nutritional value. Okay, nutrition enhancement can be done by gene addition, gene subtraction. Okay, done. Okay, next thing about gene addition is our most common example, BT cotton. How many people know about BT cotton? How many people know about BT cotton? So students, the story is simple. Cotton, very soft, pavum, okay, very pavum, simple cotton, used to always get affected by, or used to always get, attacked by insect. At one point, cotton decided that, okay, I'm too soft, I'm already getting half is falling down, half I'm falling for, half is getting, attacked, I'm getting attacked by insect. Now I'll just give up. Okay, so cotton gave up. Cotton used to continuously get damaged with insects. Now there was another bacteria called as Bacillus thuringiensis. Bacillus thuringiensis also was getting affected, but it had a self-defense mechanism. It did not give up like cotton. It was the tougher one. So Bacillus thuringiensis started releasing Bt toxin. Okay, an insecticidal. That means, most of our insects, when we are near to them to escape from the predator, they will release something, right? Same way, this Bacillus thuringiensis bacteria also started releasing Bt toxin. Okay, this Bt toxin, then scientist was like, okay, wait a minute, what if I put this Bt toxin into my cotton so that the cotton can get away from insects? Make sense or not? Okay, make sense or not? So that was the technique. Okay, Bt cotton. So what is happening, students? Basically, cotton, okay, this is your beautiful cotton. You should just agree. Okay, this cotton was always getting affected by insects. Okay, affected by insects. It could be the flies, mosquito, diphtherians, many of these insects was affecting it. And at one point, cotton gave up. Cotton was like, okay, it's not for me. I cannot, okay, I cannot really do this because it's too much stress for me. So I will just give up. But at the same time, there was a bacteria called, okay, there was a bacteria called as, okay, Bacillus thuringiensis. Okay, Bacillus thuringiensis, which for self-defense, okay, which for self-defense kept releasing, what is, okay, self-defense kept releasing what? All of you in the chats. Bt toxin. Okay, to protect itself. That's all. Simple thing, to protect itself. Okay? Yes or no? Doesn't look like cotton, huh? People shouldn't tell such rude things. I've always told you people. Yes, very much like cotton it looks. Okay? Yes. Now, what did scientists think about? Scientists was like, okay, fine. I will put this into the cotton. So the cotton can be protected from insects. Simple as that. Simple as that. Okay? That is your Bt toxin or your Bacillus thuringiensis. Now, what did they do? They call it, there is one introduction of your crygene. Crygene. What is crygene going to do, students? All of you? What is crygene going to do? What are they talking? That caterpillar is like, oh, very busy there. Okay, so crygene, crystalline gene. Okay, this crygene, students, is in the proactive form. That means it is not activated. Okay, it is in the inactive stage. Okay, it is in the inactive state. So in this inactive state, what happens if an insect consumes? Okay, if an insect consumes, then in the insect's gut, okay, in the insect stomach, inside the gut, there will be alkaline medium, right or not? Okay, in the insect gut, there will be alkaline condition or alkaline medium. This alkaline medium will basically activate, okay, activate this cry gene, and what will this cry gene do? It will go and stick to the gut of the, it will stick to the midgut of the insect, and obviously it will form pores, and that pores will lead to killing the insect. Killing the insect. Any doubts? Any doubts? Yes, for edible crops we cannot use this. It will affect. It will affect. Why will you consume cotton? Why will you consume cotton? Please tell me. Okay, why will you people consume cotton? Okay, yeah, sunlight will affect, so we will not do that kind of thing. We will basically do the gene modification, complete gene modification. Okay, so students, yes, so look here, cry gene is there. This cry gene is not cry gene, it's crystalline gene. It's inactivated. It will get activated in the insect gut because of the alkaline medium in the gut, and that will stick to the gut of the insect. Okay, this one, as soon as it gets activated, it will stick to the gut of the insect and it will form pores. Okay, it will form pores of the gut and later lead to lysis. Okay, lysis. Yes or no? Students, we are talking about insect. Why are you thinking about why are you people thinking about yourself? Stop thinking about yourself. Okay, yes, so many unnecessary doubts. Wow, wow. Something and all these people are asking. Okay, tell me which are the insects. Tell me which are the insects. Flies are there. Mosquitoes. Diphtherians. Then bollworm. Okay, so many insects are there. Okay, whichever you can remember, please do remember. Okay, lysis, khatam, kale khatam. Okay, yes. Now, students, what you need to know very specifically is about your two genes which are very, very specific. Okay, you know, all these people are very specific. They will not harm everything and anything. It is very, very specific. Okay, yes or no? Yes. So many are there. Diphtherians are there. You can write whichever you remember during the exam time. Okay, you can remember whichever you, at that point which you can remember. Okay, lepidopterans, diphtherians, you can remember bollworm. Okay, now students, we are going to learn few exceptions, not exceptions, specificities. Okay, specificities. I'm writing it in a box here. What is that specificities? All of you can think it. Cry, cry. Now they'll start crying. Cry, 1AB. Which one it will attack? Okay, so it's getting harder and harder. This solution only ended up causing more problems to people. So they were like, okay, we have to do something to increase the shelf life, okay, of tomato, and also to increase, to reduce its softness. Yes or no? Yes, yes, it will be soft. So, and also to increase its fungal resistance, because again, if you keep this tomato a longer time in the fridge, you would have seen that if it is touching to the bottom, right? We have a, if that one tomato is touching to the bottom and for a very longer time there was moisture content, that entire thing will catch fungus and it will immediately spread the entire, it's like the community. Okay, one community of people will be like, oh, I heard this, like that. This tomato will be like that. So it will spread entirely very fast. So to increase the fungal resistance, people were like, okay, this is not our solution. Spraying pesticides, weedicides, fertilizers, spraying PGRs will not help. we need to do something more. We need to do something more. So what did they do something more? Again, they used the RNA interference technology. Is this clear? Yes, gossiping. Okay. Yes. Yes. So students, they took a tomato. Okay. Na, na, na. Wow. My tomato is marvelous. Okay, students, they took a tomato. To this tomato, they see there was one person who was the villain. The villain was none other than Polygalacto. Okay, Polygalacto. So this person was the person leading to the softness of the tomato. Okay, so as days were passing, what happened? First day the farmer is very happy, or the vegetable seller is very happy. He used to sold all the tomatoes. If there were like 2-3 kgs of tomatoes, what would happen? That tomatoes would have got increased, that softness would have come. So for that, we had to gene suppression. We had to make this fellow inactive, or we have to stop its activity of increasing the softness. Okay, this was the first ever finding. This finding actually helped, okay, people to save the tomatoes. Is this clear? So what did we do? We basically silenced this gene. Problem sorted? We just silenced this fellow. So when we silence this fellow, what will happen, students? It will automatically reduce the softness, because this fellow is the one who is doing the softness for the tomato, and we don't want it because we want to keep it for a longer time. We want the tomato to increase the shelf life. So what did we do? We silenced this gene and we told that, okay, problem sorted. Is this clear? Yes or no? Quickly tell me. Okay, and this tomato was called, very fancy name, okay, flavor savor tomato. You can read on about this in Wikipedia. There's a lot of information. Okay, so much is there about this. They also silenced one more gene which is helping in release of APH. Okay, again, that is very complicated. That's why I didn't want to tell. It's aminoside phosphate. So that also was helping with the release of neomycin, which is an antibiotic. It was releasing neomycin. That also was silenced. So 2 genes were silenced in this tomato to form the flavor savor tomato. How nice, no? One fancy name it has given. Yes, clear? Yes, very good. Okay. Yes. So students, this would have inhibited. So this will basically stop it from rotting. So it will inhibit rotting. This is a little additional information, but it is good for you. So it is good. Okay, keep it. Okay, now, ready? Medicines. So we finished agriculture. We studied green revolution. After that, we studied tissue culture. After that, we studied somatic hybridization. We studied RNA interference. With RNA interference, we saw gene addition and gene suppression. Are we good to go? Everything, one example also you have understood. Now we have to understand about medicines. Okay, so medicine is very interesting because medicines can be done without biotechnology also. Why do I need biotechnology to do medicine? So many medicines are coming out every single day on the market, and do they all need biotechnology? Not required. But there are some things that only biotechnology can do, right? Now, different types of therapeutics. Not only that, I can also do large scale production. That is the best thing about biotechnology, right? I can do large scale production. That means I can treat the entire population very easily. Is that possible by pharmacies? Is that possible by people in the lab who are making medicines? No. Right? No. So I can do, with the help of biotechnology, I can make sure that at least half the population getting the medicine at the same time. Now during COVID, vaccination, how fast did entire India, get vaccinated? It was in a very short span of time, because that is how biotechnology is. That is how fast it is. So if I want to do a large scale production, I will definitely go for biotechnology. Yes. Yes, your bioreactors, you would have studied it now. Yes or no? Yes, very good. Not only that, the most important thing is not to have any immunological response. Okay? That means, students, it is very safe. It is very safe. Yes or no? Because it is happening with the help of biotechnology, it is very safe. The amount of allergies or response that people get by eating some medicines is crazy. If you just normally read some papers, research papers about how some medicines have a very drastic effect, that it leads to allergies, skin rashes, okay, so many things. My tongue is it's going, okay, I don't know why. From the time I came, it's going full some activated mode. It is going zig zig zig. Okay, so yes, so students, what is happening? This will reduce the immunological response. Okay, at the same time, it is safe and effective. It is safe and effective. So this is the reason why you will use biotechnology in medicine field. Okay, because I can do different therapeutics, also large scale production, no immunological response, and safe and effective. Fine, good job. Okay, all of you understood, right? Now tell me which is the medicine you want me to make. I will make it. Okay, because why not? So which is the medicine that we are going to make? All of you in the chat. Oh, very important one number I had to tell in your NCERT. Okay, around 30 therapeutics have been found, but out of that only 12 is being sold in the market in India. Okay, but the funniest thing is, in India there are so many medicines which are not, okay, so many, not medicines, I would say, so many chemicals which are there in the food products which are completely banned in abroad, okay, in other places. Okay, so that is very funny that out of 30 only 12 is being sold in India. But there are a lot other things like food color, other things which are completely stopped in other places, but it's still continuing in India, and small scale people still sell it, right? Okay, awesome. 30 therapeutics and 12 is sold in market. Okay, now what did I tell? We are going to, oh, you people want me to make insulin. Chalo, if you tell, I'll make insulin. Who is this insulin? Who is he? Okay, India wants to know. Who is he, students? Who is he or she? Some feminists, they'll come off with like, ma'am, how can you tell it it's he? Okay, yes, insulin. How am I going to make insulin? Who is insulin? You tell first, then I'll tell. So there is one organ, right, called pancreas. Okay, there's one organ called pancreas. All of you know that. This pancreas, okay, this pancreas have islets of Langerhans. Who does they have? They have a set of cells called islets of Langerhans. This islet of Langerhans have beta cells. All this I have taught in this channel. Okay, you can go check the playlist. All the chapters are done. All the chapters are done. Only living world and biological classification, it's happening in the crash course. Okay, so those who want to join the crash course, quickly go to the description box. There is a link. Click on that link. It is for 1,000 rupees. You will get the entire content. Even if you don't want to attend the class, you can definitely take all the Tattva books, PDFs, mock test, everything you will be able to access with this 1,000 rupees. Okay, and it will definitely boost your scores. You can use the code GHCC and get it for very minimal price. So students, pancreas, islets of Langerhans have beta cells. This beta cells releases insulin. But what happens sometimes in some bodies, right, some people's body, insulin production might be less, or the amount of glucose in the blood will have increased. Yes, now that's why this person will end up dealing with something called as diabetes mellitus. My tongue really needs one big hitting. Okay, diabetes mellitus. Okay, now this diabetes mellitus, students, why did I say it is an increase? What is happening? Yes, beta cells of pancreas. Very good. Okay, write it. here. Okay, this lack of insulin will cause diabetic mellitus. Okay, yes, diabetic mellitus. So students, understand, okay, this diabetic mellitus is not like diabetic insipidus, because diabetic insipidus is a lot of water in the urine. Here what is happening, glucose level has increased drastically. So this person, what is happening, he needs insulin, but insulin is not enough in his body, that much is not produced. So then they were like, okay, everything we have a solution, we will make who? We will make the animals, we will work on animals. Okay, we will slaughter cattle. This is the solution given. Okay, so first insulin, the formation, they thought, okay, we will do one thing, let's slaughter, okay, cattle and extract or get insulin from this, okay, this cattle. But it turns out to be a problem. This insulin which was, you know, taken from cattle ended up giving allergies to most of the people, ended up giving severe allergies. That means they understood that this has a, you know, opposite response. Okay, opposite response. Now the thing is, you should understand that slaughtering cattle gave us insulin, like we got insulin from slaughtered cattle, but this cattle says your cattle and pig also, this did not survive the need. Okay, this did not survive the need. For you to make insulin, students, what is the first step? If you want to make paneer butter masala, what is the first step for it? All of you tell me in the chat box. Let me see how many of you have cooking skills. Okay, tell me how many of you have cooking skills. No, not that. If you want to make paneer butter masala or paneer butter chicken, what is the first thing you need to know? Buy paneer. Wow, okay. Before buying paneer, before, like, only paneer is enough. You need a lot of things. So how will you know what and all is required? I'm giving you a clue, yeah, people. Tell. Tell. All of you. But these people, yeah, full recipe only they're telling. Yes, recipe. Exactly. You need to know the recipe. You at least need to know how it looks. Yes or no? You at least need to know how it looks. Okay, you will Google. You will go to YouTube. You will open YouTube and you will be like, okay, paneer butter masala, and you will look at it and you will be like, oh, this is how. I thought it was like this. Right? At least you should have an idea. Lot of the times we end up making cakes. It will not even look anything like cake. It will look like, okay, I have put some cocoa powder in water and I have mixed it and it looks like a dosa batter. So if you don't know how it exactly looks, how will you make it? Okay? Okay? So first thing you will do is open Google or YouTube and search this auntie, Mr. Cooker, say how I want to find how is paneer butter masala made. Same way, students. Here, insulin, if I want to make insulin, I should be in a position to know how insulin looks like. I should know insulin structure. Yes or no? Okay? Yes or no? I cook well. Okay? I cook well. If you people all crack Neet, I will give you food. Okay? Crack Neet and come to my home. I'll prepare food for you, all of you. Okay? That was just a thing. Don't come to my home so fast. Okay? So, yes, but I will definitely cook and give you people if you crack Neet and make me proud. Right? Yes. Now, keeping the promises apart. Now, students, understand that there was a company called Eli Lilly. Not Ali Lilly, Eli Lilly in 1983 or 1953. All of you check and tell me 83 or 53. Okay, they were like, let's make human insulin. Instead of taking it from any cattle, let's just make human insulin. So for that, the first step was to know the structure of human insulin. So let's draw the structure of human insulin. Simple structure. Two strands were there, one A and one B. Two strands were there. Okay, I'll show you 1 second. Let me finish drawing. B. This A and B was connected with C. Okay, this A and B was connected with C. Oh, I draw well. Chumma. Their kidney will look like one Rajma and they're talking to me about diagrams. Okay, so look here. Students, what is happening? All of you, don't forget who joined right now. Also, something big is coming at 7 o'clock. Big surprise, especially for the Tamil Nadu students. Okay, so you will all be excited because I am super excited to reveal it. So I'll be coming live and telling you what the surprise is. So do not forget, the session is already live. Quickly go and smash the like button and click on notify me. Okay, yes, students, what is happening? A and B is there. This 2A and B is connected with C. Okay, C peptide. Got it? I should write it outside, no? Otherwise you'll get confused. Yes, C peptide. How is A and B connected? A and B is connected with the help of disulfide bonds. Okay, disulfide bonds. You can see NCERT, same diagram will be given. Okay, disulfide bonds, it is connected. They understood, they saw the insulin diagram. They understood how insulin looks and they noticed that this one, after maturation, okay, after maturation, this insulin, okay, after maturation, this insulin is going to look somewhat like this. A, okay, B. The C will not be there. Okay, after maturation, what will happen? The C peptide will be removed or the C chain will be removed. Okay, but still they will have the disulfide bonds. Okay, do not forget that. Okay, the disulfide bonds will still remain. The disulfide bonds will still remain. So they were like, okay, now the C chain, that the C chain has already gone. Okay, C chain is out. Why will I need a C chain, right? Why will I need a C chain? So they thought very, carefully. Eli Lilly company thought, already my tongue is twisting. Upon this Eli Lilly, my tongue will twist more and I don't know what I'll tell in further. Okay. So, yes, students. So what they thought was, okay, anyway, we do not have to make the C peptide. Let's do one thing. Let's make human insulin by using E. coli bacteria. By using E. coli bacteria. Are you all with me on this? So they took, where do I write? Okay, let's draw it here, students. I'm writing it here. Okay, what did this company do that it became so amazing? Okay, simple thing they took separate, okay, A, B, they formed and they joined it together with the help of disulfide bond. Simple thing. After maturation, anyway, only A and B bands are there. Only A and B strands are there. So why to chumma waste energy in making C? So they basically took A and B and closed it with disulfide bond. Is this clear or not? Okay. So what did they do? They took A and B, inserted it into the plasmid of E. coli and they formed your insulin, human insulin. Okay. If you want to write, you can write it here, to the plasmid of E. coli. Inserted into the plasmid of E. coli and with the help of disulfide bond, they joined it together. Now they got human insulin. Now we don't have to slaughter any cattle. We do not have to worry about allergies because we got the same structure of a matured insulin. Are you all with me on this? So this is your insulin before maturation with the C peptide bond. Okay, A, B and C peptide bond with disulfide bond. After maturation, we understood that the C peptide bond gets removed. So only A and B is there. So they thought very wisely. They were like, let's not waste resources or money on making the C peptide. So let's just make the matured insulin that has A and B with disulfide bond joined together. We will put that plasmid into the E. coli. Are we good to go? Are we good to go? Yes, awesome. Awesome. All of you understood? That's all. Human insulin done and dusted. Human insulin done and dusted. Okay. So this is with C peptide. Students, C peptide chain, you can tell C chain. Okay, I'll tell you one thing. Question was asked in one of the years. What happens after the maturation of insulin? They had given some option. They gave A, B with disulfide bond. B and C with disulfide bond, A, B, C, then A, B without C peptide bond. So which will your answer be? A, B, C, D. Which is your answer? The question was, after maturation, what happens to the insulin? A, B with disulfide bond, okay? A, B, B, C, A, B, C, and A and B without C peptide. Yes, you have to answer A, B without C peptide, okay? You cannot answer A, B, if it's given like that in the question, read the question properly. But A, B without C peptide is good, okay? Good to go. This one, fine? Yes. If you understood this, let's move on. Students, next topic is gene therapy. Okay, gene therapy, I'll just fill water and come, so take a 2 minutes break. Okay, I'll just fill water and come. So my throat also. Okay, just 1 minute, I'll fill water. You also all drink water. Don't forget PP. Don't forget PP. Slides, don't forget.
Yeah, I'll upload it. How are you doing, ma'am? Have you given him a break?
No. I took a water break to go fill the water.
Oh, nice. How are they doing, ma'am? Are they able to do this chapter easily?
Yeah.
Or did they already forget what we thought in principles?
I guess they.
Nah, probably they would not have forgotten, ma'am. Students, are you able to relate the two chapters, that is principles as well as your application? Because when both the chapters you take, right, they're approximately 44 marks, ma'am.
Yeah, that's what I was telling. 2020, from this chapter, 6 questions came.
What?
Yeah, 6 questions.
Oh my god.
Sir is making me laugh, Prince, for no reason.
Okay, you continue, ma'am. I'll just go and I'll.
You want to ask them a question?
They'll know.
They'll know.
Not
Oh.
able to hear, is it?
They can, no? I didn't switch off. Yeah, I didn't switch off. They're telling hello, no?
Okay, hello. Okay.
Yeah.
Yeah, cameos, yes. Okay, you continue, ma'am.
Yes. So, I told them about the surprise. Something big is coming, no?
Told them.
No, I just tell them what it is, but something big is coming.
Well, reveal it today only, right?
Yeah, today. They're waiting. Sir is funny. Sir was very serious, We have motivated him to become funny, to show the funny side of Sir. Sir used to be like, oh, lecture, students, lecture. Yes or no? Okay, that was me imitating Sir, okay? Okay, fine. Students, here, you can otherwise write, one more thing I will just add, okay? Here, write inactive. Okay, inactive insulin. That means it's not activated yet. Here you can write active insulin. Okay, same thing, matured and same thing, active and inactive. Okay, yes, fine. Look here, next is gene therapy. Students, very, very important. But also most of the people will be like, oh ma'am, it's so difficult. I can't understand. Nothing like that. Okay, it's a very simple topic. So let's try to understand. So this gene therapy, that means with something we are modifying a gene at the embryonic age. Because of that, one kind of a condition is completely avoided. Yes or no? So this first happened in a 4 years child. Okay, 4 years old child. Okay, girl child. This happened and she had, can any of you guess what she had? Guess quickly. Yes. So, in simple words, if I want to make a another person understand what gene therapy is. It's basically correcting the genes. Okay, correcting the genes. Yes, that is basically gene therapy. Yes, students, look here. Severe combined immunodeficiency was seen in first a girl who was around 4 years old. Okay, so what was happening? She had ADA deficiency. What does she have? ADA deficiency. Isn't the chapter easy, students? Are you feeling it's difficult anymore? Everyone who was crying, you know, biotechnology is so difficult, biotechnology is so difficult. Do you find it difficult again? Yes? Yes? Yes or no, students? Are you finding it easy now? All of you, tell me in the chats. Tell me in the chats. Yes, is it? Used to cry. Used to cry. Easy, no? Yes, it's a very, like, it's a simple chapter, students, but then students have that fear. That fear is already in your head. That is the reason why you feel like, oh, no, I cannot do this. Okay, chalo, let's continue. So, ADA deficiency, adenosine, okay, adenosine, full form if you want to know, deaminase, okay, ADA full form, adenosine deaminase. This was seen in this child, okay. So, obviously, they tried different type of treatments. What could be the treatments that they tried? First, they tried bone marrow transplantation, okay, bone marrow transplant. Okay, students, now when you do bone marrow transplant, that person, that girl, right, that girl who is there, the 4-year child, she is able to form lymphocytes. That lymphocytes are helping for a little of ADA deficiency, like balancing it. But the problem is, none of this is a permanent solution. What do I mean? None of this is a permanent solution. Otherwise, one more thing I can do is enzyme replacement therapy. Enzyme replacement therapy. That means every time the kid comes, okay, and she gets an injection, okay, and that will build this lack of ADA deficiency will be maintained. Okay, every time we give an ADA enzyme, that will do ADA, form ADA, problem sorted. But how long? How long? Because whatever should be there in our body has to be there, no? Because how much ever we give from outside, none of this can be a permanent solution. All this is a temporary solution to save the child. Yes or no? Everything is a temporary solution to save the child for some period of time. Now, at the last, obviously, we are talking about gene therapy. So, we will do gene therapy also. Okay, we will do gene therapy also. So, we will, you know, the child, we will give a new lymphocyte, right? New lymphocyte. Okay, lymphocyte, gene therapy, we change the gene, new lymphocytes are formed. That lymphocytes will do ADA. Okay, okay, ADA. Now, students, what is happening? You should all understand that lymphocytes does not have a longer lifespan. All of you know this or not? We have studied human health and diseases. Do lymphocytes have a very longer lifespan? Like, okay, until this child, you know, anything happens to this child, until that the lymphocytes will be there. Lymphocytes have a very shorter lifespan. It could be 5 to 8 days. It could be 1 to 2 weeks. It could be 4 to 5 days. Now, that means every single time, once the lymphocyte stop doing his job, the child has to come back for gene therapy. Is that possible? Is that possible? Then you'll be like, ma'am, why are you telling this under gene therapy? Students, gene therapy is the solution. Gene therapy is the solution, but the catch here is that this kind of things, okay, this kind of alteration in the gene therapy should happen in the embryonic stage. Gene therapy, such kind of changes should happen in the embryonic stage. Only then there can be a solution. Only then there can be a solution. Definitely gene therapy is the best. Right? Gene therapy is amazing because you're able to correct the gene and fix the problem. But the problem here is, once you're in an adult state, or once you have, from the embryonic stage, right? Once in adult stage, we cannot do anything, because none of that can be a permanent solution. Everything will be for few days, few weeks. After that, what? Right? If you want a permanent solution, it has to happen during the embryonic stage. Is this clear? Is this clear or not? All of you tell me in the chats. Clear? Okay. So then the problem is T cell maturation is not happening, right? T cell maturation, some defect is there, okay? That is because of your adenosine deaminase, okay? T cell maturation. So that we can do with the help of a lymphocyte. But again, lymphocytes are not longer lived. It cannot stay until this person dies. So again, after 4-5 days, this person will have to come and do gene therapy. How is that possible? How is that possible? Does it even make sense? So that's why gene therapy has to do in the embryonic stage. Good to go? Good to go? Chalo, how many of you have registered for your All India mock test? All India mock test. Quickly tell me. Students, the last date for registration is April 28th. After that, you will not be able to give the free mock test, and it's happening every Saturday, Sunday. Okay, after block, why don't you write this and understand where you stand? Okay, which college will I get? Which rank will I know? Because 25 lakh plus students, still the counting hasn't got over, still the final number hasn't come. That means you can imagine how many would have registered when the second time registration got opened. How many, like again, correction window got extended. More people would have registered. More people would have, come into proper, they would have corrected the mistakes they did. So what are you waiting for? Just go to the description box, click on it, enroll, attempt it, no problem, just give it a try. Just give it a try, see where you stand. Otherwise, how will you know that, ma'am, I'm in 430, I have to make it up to 500, 600, 700. How will you know without giving a test? Right? And every single topper you open on YouTube, you open news and read, every single topper would have told, ma'am, I have practiced so many mock tests during my revision time. This is what you have to do. Now, this is the time to do. Okay? After May 5th, if you sit and cry telling that, oh, I did not practice mock test, then I don't know whom to even scold. Okay? And if there are any offline centers near your place, okay, then go right there and write and get all this free kit. Okay, you will get 360 analysis. You will also get your ranks. At the same time, you will be able to know your All India rank, and you will also be able to get some kit. They'll give you a preparation kit. Okay? Okay? Students, any doubts, you can drop a mail to Vedantu Care. Okay, that Gmail we have shared on the Telegram. Right? Okay? Enzyme replacement therapy and gene therapy is definitely two different things. No, I'm giving an enzyme to do one job. Gene therapy is what? I'm modifying the gene so that it can basically, from the roots, it can save the problems. Right? It can save the problems. So both cannot be compared. One is like you're giving paracetamol. Like when you're giving like, okay, fever is there, I will correct this. But this is what you're giving a temporary solution. You're giving a permanent solution. Okay? Yes, can we continue? Chalo. Okay, students, molecular diagnosis. So if all of you become doctors, you will be doing diagnosis, right? How awesome it is to even tell. Okay, when my students become doctors, they'll be doing diagnosis. That means they'll be checking, they'll be giving treatments, and they'll be telling what you should do, what you should not eat, what you should, everything you'll be telling to that person, and that person will believe it like the way they swallow the tablets. They'll be like, yes. Now, example, Dr. Jay Ganesh have told me this, I will definitely follow because he is knowledgeable, he knows everything, and I will continue. Yes. So, that kind of a trust is built in you when you diagnose and give a treatment. Yes or no? So all of my doctors, all of my future doctors, I just pray and hope that all of you become doctors, all of you are in your MBBS next year at the same time when I'm teaching biotechnology, because the next batch of students will be crying and I'll be teaching them, I'll be scolding them like this only. That time I hope all of you will be like, hi ma'am, I'm in this college, I'm in this college, right? That's the most precious thing. Nothing else is more precious. So that time, students, you will be doing diagnosis, right? Every time you'll be doing diagnosis. You'll be like, okay, do you have cold? This is what you should eat. You have fever. This is what you should eat. Do you have a stomach, abdominal pain? Maybe this is the condition, right? So that is same thing is called diagnosis. That means you have to understand how to treat a disease. All of you tell me, all of you know something or the other about this, right? So all of you tell me, how do we do the basic test? So the basic conventional method, that means the olden days method, or the, actually to be frank, the basis, now also we do this, is urine test, right? Blood test, serum test. This is a conventional method. Okay, that means before even technology has developed to such a level, we used to do this all the time. Now we have modern techniques, right? Now we have some modern techniques. What are the modern techniques? First one is recombinant DNA technology. Students, I fell, so my hand is, it's very painful, okay? Because I fell, it was, the pain hasn't gone, I haven't given it time. Recombinant DNA technology. Mark happened in this hand, I supported with this hand. So my both the hands are damaged basically. Okay, so we have a damaged team, that is me and Diksha mam and I. We are the damaged team, okay, fully damaged, everything is damaged, neck is gone, hand is gone, okay. So yes, so mark came in this hand, but support, like I kept this hand somewhat, okay, so that's why I'm not able to write. When I'm lifting my hand, it's paining. Okay, so recombinant DNA technology, modern technology, then your PCR, okay, and ELISA. Okay, enzyme linked immunosorbent assay and PCR polymerase chain reaction. Now students, on what basis does your ELISA happen? Which is the enzyme that is used? Enzyme? Anyone can tell me which is the enzyme it is used? All of you, phosphate, something to do with phosphate. Okay, phosphate. That's why I'm taking a break sometimes, so that my hand just, you know, just feels light. Yes, tell me. Alkaline phosphatase. Yes or no? Okay, if you don't know also it's fine, but just like in case somewhere you happen to read or something, alkaline phosphatase. So students, this is conventional technology, that means urine test, blood test. But for this people to show the result, urine, blood, serum, that means the bacteria or the pathogen should be in higher concentration. Agreed all of you? This has to, if this has to show result for me, that means my pathogen, okay, my pathogen concentration has to be very, very high for me to show the symptoms and for me to get the sample value. Yes or no? Now, at the same time, if you see here, there are techniques like PCR which I can use for amplification, right? Amplification. This techniques will help me to understand even when the pathogen is in a very lower, you know, concentration. If it's higher, PCR cannot detect. So if it's in lower concentration, also I will be able to amplify and get to know what is happening. Yes or no? Agreed all of you? Yes? Yes or no? All of you tell me. Yes? Fine. Conventional method and modern method. So the difference you can understand. This one we only go for a blood test or a urine test when we start seeing some symptoms, right? You have an urine infection. You're not been drinking enough water. How do you know it? You start getting abdominal pain, right? And when you do micturition or urination, you feel the pain in the abdominal area. You feel like there's a lot of pressure. Because of that, you go and give your urine test. At the same time, if you have a like, you know, you see a small swelling in your throat area, or you see that you suddenly started putting on a lot of weight, or started suddenly decreasing weight like 10 kg, 15 kg, you'll go give your blood test to see if there is any thyroid or anything, diabetics. So that is only when the symptoms are in that level. That is because the pathogen has multiplied in your body. But at the same time, you have modern techniques, even with the smaller amount, we can amplify and see. Okay, we can amplify and see. So let's study from NCERT only, students. Look here. Presence of pathogen, be it bacteria or virus, is normally suspected only when the pathogen has produced a disease symptom. Agreed? What did I say? Same thing is told in NCERT. By this time, the concentration of the pathogen is already very, very high. Okay? Pathogen concentration is very high in the body. However, very low concentration of a bacteria or a virus at the time when the symptoms of the disease are not yet visible can be detected by amplification. Got it? By amplification. Okay? Amplification of the nucleic acid by PCR, polymerase chain reaction. Can you explain? Students, your job. PCR is now routinely used to detect. First job of PCR, first ever function of PCR, to detect HIV in AIDS patient. Okay, so HIV in suspected AIDS patient, it is being used to detect mutation in genes in the suspected cancer patient. So also in HIV AIDS patient as well as in cancer patient, PCR is being used at the same time to find other genetic disorders. So three main functions of PCR. Let's write it here. First function, all of you tell me. 1 second, students. Maybe if I stand on this one, I can write it. My hand won't pain. Okay. Yes. Students, so what is happening? First one is HIV. Okay. That is to find the HIV virus in AIDS patient. Okay. Second thing is what? Second thing is what? Cancer patient, to detect in cancer patient. Okay. And third one is what? Third one is to find any other genetic disorders. Don't worry, all these notes you will get on Vedantu Neet English Telegram and Biology by Gopika, both the telegrams, you will get all these notes. Okay. Chalo, this is the function of PCR. Now, look here, a single stranded DNA or RNA tagged with radioactive molecule, that is called a probe, is allowed to hybridize. So students, here they are talking about autoradiography, which we studied in your DNA fingerprinting. Okay, autoradiography. Okay, autoradiography. This autoradiography, students, last time also I told you, we use a probe. What do we use? We use a probe. That is a single stranded DNA or single stranded DNA. Now what is going to happen? Yes, identify other genetic material. Okay, we use a single stranded DNA, we can use a single stranded RNA, and what are they going to do? This autoradiography means what? I found a band. Okay, this probe will be complementary to this. It could be a single stranded DNA. This will go and bind with this, and this will be tagged with radioactivity. Okay, this probe will be tagged, okay, with radioactive material. Yes, we studied this in DNA fingerprinting. How many of you can remember? How many of you can remember? Okay, it will be tagged with radioactive molecule. Now what will happen, students? This probe will go and bind with the normal genes, not the mutated one, not the affected one. It will go and bind with the normal. Now when I see it under the radiography, when I see it under the autoradiography, I'll be able to see the strands which are normal. Yes or no? I will not be able to see the ones are mutated or the ones which have problem. That is what they're talking about. With this knowledge in your head, let's read this paragraph. A single stranded, what did I tell? A probe will always be a single stranded DNA or a single stranded RNA. Okay. A single stranded DNA or RNA tagged with a radioactive molecule is allowed to hybridize to its complementary DNA in a clone of cells, followed by detection using autoradiography. The clone having the mutated gene will hence not appear. Do you understand? The clone having mutated genes will not appear. Why? Because the probe will not go and bind. Probe will only go and bind on the normal gene. Is this clear? Good to go? Okay. And will not hence appear on the photographic film because the probe will not have any complementary, like the probe will not go and attach with the mutated genes. ELISA is based on the principle of antigen-antibody interaction. Infection by pathogen can be detected by the presence of antigen like protein and glycoproteins, etc., or by detecting the antibody synthesis against the pathogen. So basically ELISA works on antigen-antibody interaction, and here small paragraph about autoradiography. Now what happens when students are studying? No, they will ignore this. They'll be like, oh, autoradiography, what will come? But the question can come, is the probe made up of double strand, single strand, RNA, DNA, protein? Any kind of question can come. Yes or no? NTA exactly knows which one you will skip. That only NTA will give question. Chalo, last topic, not last topic, last main topic of this chapter is transgenic animals. Okay, transgenic animals. Students, if you want, we will just write it here. Okay, here, let's just write, one second, what we just revised. Okay, ELISA is basically working on antigen antibody interaction. Okay, yes. Other than that, what did we study? We studied about autoradiography. Okay, autoradiography. I am writing this notes because some students reads my note but they do not read the NCERT snip. So I thought I'll just write it so it's safer. Okay, autoradiography, students, what is there? A probe is there. Okay, a probe is there. That probe should always will be a single stranded DNA or a single stranded RNA. Okay, and they will be complementary and they will be mainly tagged. Okay, they will be tagged with radioactive molecule. Okay, radioactive molecule, and what will it go and bind? It will only bind with the normal genes. Okay, normal genes, not with the mutated one. Okay, not with the mutated one. Clear? Okay, so probe, yeah, that's all. Okay, normal genes, not with the mutated one. There's no place to write, but yeah, I hope this is clear. I cannot write it there also. Chalo. Okay, here I've marked anyways. Now, look here, students, transgenic animals. Transgenic animals means animals which have been manipulated, whose genes have been altered or manipulated. And the biggest example is our mice. Okay, biggest example is who? Mice, because that Pavam has sacrificed his life entirely for molecular biology, biotechnology, all the fields of biology. Mice has sacrificed its life. Okay, so transgenic animals are animals that have their DNA manipulated to possess and express an extra foreign gene. That means basically, if I want to know what would be the reaction if cancer, if this medicine was given or if this gene is altered, I will inject it on a mice and I will see the result. So don't disrespect mice, give respect. Okay, are known as transgenic animals, right? There's a statue also for mice. Now, transgenic rats, rabbits, pigs, sheep, cows, fishes have been produced, although over 95%, okay, important, 95% of your existing transgenic animals are mice. Okay, do you see? Should respect. Okay, now, students, look here. Let us try and explore some common reasons. Why are we doing this? Okay, first thing is normal physiology and development. Transgenic animals can be specially designed to allow study of how genes are regulated. Okay, how genes are regulated. Remember your gene regulation, gene expression, RNA processing, all this you studied in your molecular basis. How did all this come to a finding? How did how did Griffith do his experiment? How did Avery, MacLeod and McCarthy do their experiment? How did all these people do their experiment? Because of your power mice. Okay, so how they affect, okay, how they affect the normal functioning of the body and its development. Example, study of complex factors involved in growth such as insulin-like growth factors. This is what we want to check, okay? This is what we want to check. By introducing genes from other species that alter the information of this factor and studying the biological effect. Whatever I am marking in circles, it's important, okay? That results. Information is obtained from their biological role. So first thing is to understand the normal physiology and regulation of genes. How do they normally work? How do they get regulated? What are the biological effects? What will happen if there is an insulin factor? All this we will understand with the help of transgenic animals. Are you all clear so far? Are you all clear so far? Any doubts? Did you all smash the like button? Okay, students, look here. Study of diseases. Obviously, transgenic animals are the first people on which we will work and learn about diseases. So, look here. Study of diseases. Many transgenic animals are designed to increase our understanding of how genes contribute to a development of a disease. That means, how does this happen? If a person is cancerous, what are the things that are changing in his body or her body? What is happening? What are the genes actually directly getting involved? Does it have anything to do with my ancestors? All this connection, who is going to give? Transgenic animals, because we cannot try. We cannot do gene suppression, gene addition or RNA silencing just like that in our body, because our body will collapse. So we can only do it on animals, okay? These are specially made to serve as models for human disease so that investigation of new treatment for diseases is made possible. Today, transgenic models exist for many human diseases like cancer, cystic fibrosis, your arthritis and Alzheimer's. Students, very important question. They can ask you, for which and all disease are there transgenic models? Your answer should be cancer. Okay, I'll write it here. So again, your eyes goes here. Okay, transgenic models are there for, okay, cancer, cystic fibrosis, rheumatoid arthritis, and last one is Alzheimer's, right? Alzheimer's, yeah. So for all this, we have transgenic models. So do not skip even the small portions, okay? Do not skip the small portions. So yes, students, it's very simple to remember. AC, okay? 2 ACs, if you remember, you're good to go. Arthritis, your cystic fibrosis, again, Alzheimer's and cancer, okay? Now, biological. So second thing we understood, study of diseases. First one was normal physiology and regulation. Second one is study of diseases. Third one is biological products. Okay, very important. Medicines required to treat certain human diseases can contain biological products, but such products are often expensive to make. So that is a full stop there. Transgenic animals that produce useful biological products can be created by introducing of a portion of DNA or gene which codes for a particular product such as human protein. Okay, human protein, that is your alpha-1 antitrypsin. Yes, understood? That means when we cannot, memory loss. Alzheimer's means memory loss. Okay, students, what happens? It comes basically when a person has old age, or in some cases when they have undergone a severe trauma, or they have seen something that they cannot accept. Sometimes because of that, that person could, go into Alzheimer's. But okay, now students, see, when I want to deal with emphysema, all of you know emphysema is when your alveoli surface layer reduces and there's no more, because of continuous smoking. During this kind of situation, I want to, induce this protein into my body because my body is not making enough. Okay. So during that time also I can use transgenic animals. Is this clear to all of you? Yes or no? So I will do what? I will put this on, like, I will induce it on sheep, and the sheep will do the rest of the job. So look here, it is used to treat emphysema. Similar attempts are being made for the treatment of, again, PKU, that is your phenylketonuria, and cystic fibrosis. In 1997, the first transgenic cow, Rosie. Who doesn't know Rosie? Rosie is like among us, right? Everyone knows about Rosie. So the first transgenic cow, Rosie. Okay, it is Rosa, I guess. Produced human protein enriched milk. The milk contained human alpha lactoalbumin and was nutritionally a more balanced product. I will write this now. Okay. So much I've put, students. 1 second. Okay, let's write it. What did we study now? Biological products, right? Oh no. I'm writing with so much difficulty and the board is playing with me. Since 3D I could make. Okay, first one was what? Ready? Why is this board scaring me? Who drew this? God, what am I seeing? Okay, 1 second, students. Yes, I don't know why this board is not behaving well today. Okay, let's start. First one, what was it? All of you tell me. Yes, emphysema, right? No movement. Students, all of you, mark, put love in the chat box, put fire emojis in the chat box, show some energy. What is this? All are sleepy. Okay, so students, to deal with emphysema, which is again caused because the person is continuously smoking and reduce the surface area. So for emphysema, the human protein was alpha 1 antitrypsin. Yes, so this I want to add it into a transgenic organism. So into a sheep, again a transgenic organism. This sheep, okay, the name of the sheep is Tracy. Oh, sorry. Okay. Very good. Now little I feel like, okay, not everyone is sleeping. Okay, so students, antitrypsin, okay, a protein, emphysema. To deal with emphysema, we need alpha 1 antitrypsin, which is not there, which is very less produced in a person who is smoking. And because of that, I'm using the help of a sheep, and that sheep name, this becomes a transgenic sheep. Okay, I don't know, I keep telling sheep. Okay, transgenic sheep, which is called this boy's, I don't know, Tracy. Sheep name is Tracy. Okay, who is named Tracy. Now, crazy, not crazy students, Tracy. Now, the next person was, I wanted, okay, not I, like everyone wanted a milk with, protein called as alpha lactoalbumin. Okay, alpha lactoalbumin. So, the milk should add alpha lactoalbumin. Okay, for that, I'm going to use a transgenic cow. Okay, I'm going to use a transgenic cow that will do that job for me. So this is the biological products. Okay, transgenic cow, and the cow name was Rosie. And this Rosie gave milk which was rich in alpha lactoalbumin. Problem sorted. Understood? Understood or not? So first thing is emphysema. Okay, first thing is emphysema. Second one is milk with alpha lactoalbumin. Okay, we use Tracy and Rosie. Tracy, Rosie. Clear or not? Tell something, students. I'll only sleep off if I have students like this in class. Tracy, Rosie. Yes, this gene was added. Again, here also gene was added into the sheep. Okay, fine. This is your entire biological use of your transgenic animals. Okay, transgenic animals giving us some benefit. Okay, animals in biological product. Okay, done. Now look here, vaccine safety. This also obviously you people know that we test everything on the transgenic animals. Transgenic mice are being developed for the use and testing of not only vaccines, even the makeup items. In the makeup items, if you have turned and saw, it will be like animal tested. Shampoos, animal tested. Who is the animal? All these people only, transgenic animals. Okay, so transgenic mice have been developed for the use of testing the safety of vaccines before they are used on humans. So transgenic mice have been used to test the safety of polio vaccine. Okay, it was successful and found to be reliable. They could replace the use of monkeys to test the safety of batches of vaccine. Even monkey was tortured a lot in the prior, but lot of cases came, lot of people came in front telling that you cannot, okay, they are our ancestors or they have something similar to humans. So that's why monkeys have been reduced, because are used as transgenic. Now chemical safety test, again this is exactly what I told. This is known as toxicity and safety testing. The procedure is same that is used for testing toxicity for drugs. Transgenic animals are made to carry genes which make them more sensitive to toxic substance than the non-transgenic animals. Understood? So the animals which are transgenic will be given a gene which will be very, very sensitive to toxin. Okay, but the other one will not be that sensitive. Okay, so they are then exposed to the toxic substance and the effects are studied. Okay, because we humans do we have that much of toxic, you know, we cannot sustain that much toxicity. So for that they will make sure that they give them a gene which is sensitive, because most of the, you know, animals are not very sensitive like how we are sensitive, right? So for that they have to give more sensitive to toxic substance. Okay, carry genes. Okay, all this is like story, students, just read like story and understand. Okay, now the last topic is ready. Last topic, ready for last topic? We are done with this chapter. No more you will cry, biotechnology. Okay, no more you will cry, biotechnology. Ready, steady, go. Another 5 minutes, we'll be done with the chapter. Tell me, ready or not? Okay, so students, here we are talking about the ethical issues. See, we do find a lot of things, and with those things, how, like, will there be any ethical issues? That is what we are talking about. Definitely ethical issues are there. I'll tell you an example. Here they have given an example of rice. We know that basmati rice, so many, around 27 varieties we have, and India alone has around 2 lakh of varieties of rice. Okay, now what happens, because I am finding rice in India, and imagine Meghana or Subhashni is in abroad. Okay, and like say she's from UK, and then we have Sanvi from Canada, then we have who is that, Priya from some other place, so from Antarctica. Now all these people have found the same basmati rice. I found it first. Okay, I got patent. Okay, but UK, Subhashni changed or modified a little and she got patent there. Sanvi changed it, she got patent. Megha changed it, she got patent. Now what has happened? It causes a lot of anger in me, right? Because I found the original, I found the exact. They did slight modification and they also got patent. And getting a patent is not easy. It is like a big seal, right? It's like a big achievement that, okay, we found something and that patent is only for you, right? So when that kind of a thing happened, anger built up in people, telling that we should have a system, no matter, be it India, be it UK, be it in any corner of the world, if one person is finding it, we all should have a database of it, because when that person is trying to put a name or the family or the characteristics of it, immediately it should appear. Okay, this has also happened in your botanical aids, which has been removed. No, when herbariums used to be found, everyone, like multiple people used to find the same plant and they used to write in their own way the characters, their own way of labeling it. That is why they came up with your IUPAC, that is your, in your this one, your IUPAC, how you have, no, for all this IUPAC, your, why am I not getting the name, ICBN, all these people came up with the norm telling that no matter who is finding what, it will all come under one roof. Okay, so here you can see about basmati. So students, look here, company got patent. That means your American company got patent for basmati rice through a US patent and trademark office. Okay, understood? That means Subhashni, Sanvi and Megha got the same patent like me, but they modified it. Okay, they modified it. This caused a lot of anger. Okay, now this allowed the company to sell a new variety of basmati in the US. Not only they get patent, they also make it commercialized, right? They get money for it. No, US and abroad. This new variety of basmati had actually been derived from Indian. See, derived from Indian farmers variety. Clear? Understood this? This is what ethical issues means. Okay, this is what ethical issues mean. Indian basmati was crossed with semi-dwarf varieties and claimed as an invention of novelty. That means with a small change, like how Mendel did, no? You're round and wrinkled and round and green. Like that, with a small change, they were able to find a new variety, and that cannot get a patent because that's not the original. Original was already found, a small modification was changed, right? Okay, now if I put makeup and come, will I not become Gopika? I'll be still Gopika with makeup. If I'm not putting makeup, it will be Gopika without makeup. That's all. How can I be like, okay, Gopika with makeup, Deepika? I cannot be like that. No, I have to have my originality should be there. Yes or no? Can I become Deepika Padukone because I put makeup? Obviously not. You people tell Rashmika and some other girl's name also you people tell. No, Rashmika and one more girl's name you people tell. I look very similar here and there. So just because I put makeup, can I be called them? Can I go and act tomorrow? Not possible. So same way is the small modification, small makeup, right? Genetic makeup has been done. The patent extends to functional equivalence, implying that other people selling basmati rice could be restricted. Other people selling basmati rice could be restricted by the patent. Understood the use of patent? You can even stop them from trading. Now, several attempts have been also made to patent users, products and processes based on Indian traditional herbal medicine. Example, turmeric and neem. Turmeric and neem also, it was sold in such a way like, okay, this can cure all the antifungal issues. Yes, turmeric can cure, but who gave the patent? It is Indians. Indians found this from the ancestral times, that means from the tribals. They found that, you know, mixing turmeric and mixing neem and applying on the wound was a heal, but these people started making it commercialized. Again, problem. Okay, if we are not vigilant, we do not immediately counter these. Okay, counter these. So we have to be very vigilant. That means the entire Indian system has to be very vigilant, understanding that okay, this has been sold here, this is not the patent, it has been found from us, right? Your homeopathy and your allopathy, all this has started from ancestral times. All that has to Ayurveda, basically Ayurveda, all that has come over time, right? So that patent we only need to get. How many of you understood? Very simple. It's like just about basmati rice, and you have to know about 2 lakh varieties we have in India. Okay, 2 lakh varieties we have in India. Rice alone, India alone. Okay, in India alone, and out of that around 27 varieties of basmati rice we have. Okay, again, all this come under our patent. Okay, basmati rice. Yes or no? Got it, students? Now, most important topic, biopiracy. Let's see what is it. Biopiracy means, students, imagine you are an Instagrammer. Okay, you are an influencer. Sorry. Okay, you are an influencer. Wait, I didn't tell you about this one, no? 1 second, students. I'll also write this. Who finds all this? Your GEAC. That is genetically engineered approval committee. It's given in the starting of your textbook. Approval committee. Okay, they will give approval. Chumma, you cannot find something and be like, oh yeah, this is the cure for cancer. That's not possible. No, you have to prove it. So this people will prove it. Okay, approve it. Okay, students, imagine you're an influencer. Okay, and you got an ad, like you got a paid promotion. Okay, telling that if you show this in your video, we will pay you this much of amount. Imagine we'll pay you 25k if you're wearing this one of our dress and one of our earring. This is what was done. Okay, this is what was told to you. All of you think of yourself as an influencer. You will agree to it. You will make the video because they'll be like, ma'am, we need the video to go today itself because the stock has come, blah, blah, blah. So you will do immediately video. After doing the video, they will block you. Imagine they will block you, they are not ready to pay, and they will take that video and put it on their page, and also from your followers, lot of the people would have purchased this. Now who benefited? The dress shop people or the clothing brand benefited. Who got, who did not get benefited? You people did not get benefited. That is called as biopiracy. That means you take something and you do not pay for it, or you do not give credit for it, is exactly what happens. Now look here, biopiracy is the term used to refer to the use of bioresources. So it's happening in biological, society, right? So same way. See, bioresources by multinational companies, okay, and the other organization without proper authorization from the countries and people concerned without compensatory payments. That means people using other things which are like maybe like things from multinational companies, or without authorization. There will be companies which actually sell products which have been found by different people without giving payments. That is called as biopiracy. Is this clear? Okay. So biopiracy is the term used to refer to the use of bioresources, okay, by multinational companies and the other organization without proper authorization from the countries and the people concerned with compensatory payments. Okay, imagine you found something that would help all the industry people to cut down on electricity, or enzyme use, or gene therapy, or to forensic reports. So when there is a forensic lab, imagine there are people there are people who you have given them a setup. You're given them a setup that would actually cut down on their expense, cut down on their entire equipments. One machine is enough to find the fingerprinting, to do everything. And imagine that came in market, they are buying it, or imagine that came from somewhere and they're using it and not giving you any credits for it, or not giving you the payment. That would be, or making the first copy of it. Right now all the brands have first copy. Now some of the people, what they do on online medium, they sell the first copy in the name of original copy, and they put the price in such a rate that people believe that okay, this is the original copy. They make everything like the original one and they sell first copy. So who can give a case? The actual company can give a case on these people, telling that they are selling our products as first copy and misleading people. Understood? More than the second order example, students. Now, the Indian Parliament has recently cleared the second amendment of Indian Patent Bill. Okay, so they're improving on the Indian Patent Bill that takes such issues into consideration, including patent terms like emergency provision and research and development initiative. That's all. Chapter done. Done. Any doubts? Any doubts? Any doubts? Tell me now. So much we studied. One chapter is done. Dusted. You want to check your NCERT once? So students, I told you 3 topics this chapter has. Okay. Biotechnology in agriculture. Okay, biotechnology in agriculture. Okay, biotechnology in medicine. Okay, and then your transgenic animals. This is the headings of the topics, yes? Okay, so you can expect maybe 2 questions from this, 1 question from this, and 2 questions from this. Okay, in this you have 2 questions that could be one from tissue culture and one from somatic hybridization. Okay, so we are done and dusted, right? No doubts? All clear? All clear? Chapter has become easy? Okay. Let me also just check my NCERT. If we missed anything, added information, only added information will be there because other things are done from NCERT. Hiding, somatic hybridization, golden rice done, pest resistant done, crygene done, tobacco plant done, gene silencing, gene subtraction done. 1 second, students. NCERT is God. You have to refer NCERT. Be it teacher or student, we have to refer NCERT, no? Yes, done. Done. Okay, so I have not added questions. I have not added questions. Okay, because again video will become too long. We can do questions separately. Don't worry, we'll do questions separately. So thank you so much for joining. I asked you a lot of questions in between the class, so rapid fire also got over like that. Okay, so yes, biotechnology is over. So now when are you going to come back to the channel? You'll come back at 7 p.m. because something exciting is going to come. You will all be mind blown. So quickly share the link with all. Okay, not harmful in humans. Okay, because of the presence of acidic pH. Acidic pH in the gut. Okay, got it? That's the reason. But again, okay, that's the reason. But again, Bt toxin as a insecticide. Okay, that's the question. Insecticide, if it is sprayed on plants, what will happen? Okay, students, thank you so much. See you all. Tata, bye bye. Join back. Join back. Okay, join back at sharp 7. Do not forget to like, share, and subscribe to the channel and comment. Okay.